With that, we officially start our main event. There will be three topics for this session. For the first session, we will have Advancing One Health, Veterinary Perspective on the Latest Trends by Professor Dr. Fedik Abdul Rantam, Doctor of Veterinary Medicine.
After that, there will be a lecture on food, animal health, and production for global food security by Prof. Erdogan Mamili, Doctor of Veterinary Medicine, Master of Science, Doctor of Philosophy.
Next will be the topic of pharmacokinetics and toxicokinetics, implication for drug development and safety assessment by Dr. Akson Saintin Chai.
Without further ado, allow me to welcome Yuliana Puspitasari, Doctor of Veterinary Medicine, Master of Science, Doctor of Philosophy, and Suryo Kunchorojati, Doctor of Veterinary Medicine, Master of Veterinary, Doctor of Philosophy, as the moderators for our first session. To Dr. Yuliana and Dr. Suryo, the time is yours.
Good morning, everyone, and Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, my name is Juliana Puspitasari.
And my name is Surya Kunchorojakti, and we will be a moderator for keynote speaker sessions. Before we get started, I would like to inform all the participants that during these sessions, you will have the opportunity to submit text questions to today's keynote speakers by typing your questions into the chat box of the control panels. You may send in your questions at any time during the presentations. We will collect the questions and address them at the end of the presentation of each speakers.
In today's session, we have three distinguished speakers, and these three speakers are Professor Dr. Fedik Abdul Rantam from Faculty of Veterinary Medicine, Universitas Erlangga, Surabaya, Indonesia, Professor Erdogan Memeli, DVM, Master of Science, Doctor of Philosophy from Prairie View Icvmhs University, Texas, USA, Dr. Akson Saintin Chai from Kasetsart University, Thailand. Without further ado, let's welcome our first speaker. Our first speaker is Professor Dr. Fedik Abdul Rantam, but before that, let me introduce our first speaker. Professor Dr. Fedik Abdul Rantam, currently he is a professor of virology and immunology, Division of Veterinary Microbiology, Faculty of Veterinary Medicine, Universitas Erlangga, and also as a deputy of the Institute of Tropical Disease, Universitas Erlangga, East Java, Surabaya, Indonesia. For his educational background, he finished his Doctor of Veterinary Medicine from Faculty of Veterinary Medicine, Universitas Erlangga, and then he continued his postgraduate study from Freie Universität Berlin, Germany, receiving Master and Doctor of Philosophy in the field of virology and immunology. For his research interests are virology, immunology, and stem cell, and he produced many international scientific journals in a related field, and he has published 93 papers Scopus Index and 8 books in virology and immunology. And currently his Scopus H Index is 22. In this session, he will deliver the topic entitled Advancing One Health, Veterinary Perspective on the Latest Trends. Ladies and gentlemen, please welcome to Professor Dr. Fedik Abdul Rantam.
Yeah.
Hello, Prof. How are
Hello.
you?
Morning.
Good morning, Prof. So how are you,
Yeah.
Prof?
Yeah, fine, thanks.
Yeah, Prof.
You
have,
too, yes.
yes, Prof. You want me to help to share your PowerPoint, or you want to share by yourself?
I want 2 minutes. I want 2 minutes by myself.
Oh, by yourself. Okay, Prof. You have 35 minutes presentation time, and then we'll be continued with Q&A session. Okay, Prof. Are you ready?
Okay.
Okay, Prof.
I
The
want
time
to
is
share my
yours.
PPT. Yeah. Okay. It's okay. Yeah. Good. Moment.
Prof, can you share screen, I mean like in display full
Okay,
screen?
like this. Yes, okay.
Not yet.
Not yet.
Oh yeah, yeah, already, yeah.
Ready. Yes. Okay, thank you. Thank you for the to the moderator to introduce my CV, and Assalamualaikum warahmatullahi wabarakatuh, dan salam sejahtera. And the first honorable to the our dean, Professor Mirni Lamid. Thank you for inviting me, and also organizing committee to I am to meet you today. I will meet and share my title. Actually, One Health, current perspective and future directions. So this is a title maybe still in our thinking about the future to problem solve about the One Health system in Indonesia, of course, and in the world. So today my paper so divided into four kind about the One Health what and why actually. One Health system, One Health implementations, One Health implementation challenging, and One Health future direction. So what is actually One Health is be important in these years and maybe past 10 years ago, beginning of the outbreak, avian influenza, for example. But right now, so going to the new perspective, maybe because avian influenza right now spreading already to the bovine and not only to the poultry and cat, but other animal is possible to infect of the can infect of the avian influenza. Special SPI is 5N1 in bovine herd occur in US with virus spread within bovine herd infectious body and net. So this is a very important thing information about the how like pathogenic avian influenza has so far been detected. Sorry, in cattle 145 cattle herd and for farm worker in the same state across in United. So this is the first situation, but we don't know. After that, maybe can spread to the other country is possible. So this why one net approach is very important thing to make or to get to problem solve about the infectious disease. So right now, in 2004, so many kind of disease. Mostly zoonosis disease, not only from virus infected, but bacteria and parasitic also, and multi-drug resistant about the tuberculosis, for example, malaria also is very important thing, information how to prove some of the One Health approach to avoid of this disease. So like SARS-2, 22, and S5N1, 2004, and etc. And also there is some kind like dengue, Marburg, Ebola, etc. Right now, after that SARS coming to the Mars, for example, is maybe influence of the influence from the climate change. Yeah. And avian influenza, each one like N1, for example, a total confirmed cases about the 60 million, 60.8 million, and 17,798 dead cases. So then coming MERS cases, total confirmed about 2 million 495, etc. And also it's 5 and 9, for example, and COVID-19 also total 2 billion 708 450. So this confirmed cases is the COVID-19. Look at this one. Before COVID-19, I think you know about SARS, MERS, and then coming SARS-CoV. What is happen in the future? So this is a very important thing, the infectious disease and where and how in the distributed model actually in the epidemiology in the world. This is a very important thing. Cases, yeah, the important information about the cases and the what. This is a very important how actually the situation are emerging, researching, for example, maybe new emerging cases, not only in the SARS-CoV-2, maybe in the future is coming, for example, Hendra virus, Nipah virus is possible. And of course, avian influenza still distributed in environment. So you know that 75% of new diseases most like zoonosis. So not only causes by coronavirus, they are in animal, but you know that anthropology factor related to the change and the pathogen circulation increases contact with new animal species, including domestic species too. And the COVID-19 pandemic highlight the risk of the reverse of zoonosis, resulting from the intensive circulation of SARS-CoV-2 in the human transmission to the new animal species and circulation in some animal. There is proper highly susceptible, like hamster, minks, and wild rat relative. So this is background actually how in the important thing and the one system to lead of to protect of the health in the human. So the first global change maybe causing the climate change, led to change lost biodiversity extension. The first step maybe how the new variants about the disease coming on and then travel and then spill over to the human. Like this one from um, some kind like camel, pork, and chicken, like birds and etc. Is maybe spill over to the human, and then human to human coming to the other animal again. The serve. This is a model cycle. I think very important to know about the infectious disease and the new emerging and re-emerging disease. More is more important thing. So this one animal, not animal disease or animal zoonosis, causing the tropical disease like that. For example, some kind of tropical disease, dengue virus is still becoming problem and bigger problem, I think, in the public. And also antimicrobial resistant traits, for example, medicinal resistance tuberculosis, very important thing. You know that in different country, saw that many kind of problem with antimicrobial resistance. So from the 5% until more than 80%, like this one. This is example in the Middle East, for example, is higher than other. Like this one in India, for example, Bangladesh, also including Indonesia. So be consider about the antimicrobial resistance in the future. So one health and why actually one health concern of first appear after studies on zoonosis, according to the One Health Highlight Level Expert Panel with CPLP. This is One Health is a unified and integrated approach that aim to balance and optimize the health of people, animal, and environment in sustainable manner. So this way, this is understanding of the health of people, livestock, domestic, and also in wildlife. The mean actually one is divided three kind like human, animal, environment. How the harmonization for three pillar like this one, and the communicates, the supporting by communication, coordination, and then capacity building, and then collaboration, etc. And then coming like policy how to implement it of the one net. Actually, so needed politics, yeah, policy, resources, sector and society, I think very important to support to leading on the going to the One Health system in the world. So there is some kind the quadripartite One Health joint plan of the action, a quadripartite organization so address health issue in the human, animal, and plant environment enterprise. This is many organizations support how to deal of the to bring up the One Health to be like to solve of the problem by public health. I think a very important thing. So that's why the both organizations like WHO, FFO, UNEP, and WA is more integrated, bring integrated thinking about how to bring of the One Health system to implement it in the world is be very important thing. So One Health implementation, how it is actually relevant multi-sectoral stakeholder, for example, representation with different expertise and perspectives. This is organ of the One Health. The main sector actually human health, animal health, environment, also agriculture and culture, wildlife, plant and food, and also other sector like industry, privates, education, academic research, and etc. Disaster management, also depend on security, local government. So this is a very technical how to bring up the implementation of the One Health system in the world, and every country maybe needed the leading bring of the One Health to prove of the solve for the infectious disease like this one. Sorry. One of the implementation in it is, that based on the presidential instruction number 4, 2019 concern increasing capacity and preventing, detecting, responding to disease outbreak, global pandemic, and nuclear biology and chemical emergency. And the secondly, Minister Home Fire Regulation number 101, 2018 concern basic services technical standard or emergency disaster some affair minimum service standard. Third, finisher of health regulation given number 1501, 2010 concern certain type of infectious disease, and also Minister of Agriculture concern determination priorities on this regulation and coordinating Minister for Human Development and Culture in 2022. So like this is we want like this one. There is a harmonized between animal, human, and environment. So before like this, and then coming maybe how to improve of the One Health implementation, and then becoming like this one. This is our and their future. So, for example, in the infectious disease detection and surveillance project, they strengthen involved system capacity, rapidly track and respond, priority infectious disease such as tuberculosis. You know that Indonesia is number 2 in the world cases of tuberculosis. So this is a very important thing. So that's why we have developed about the city One Health actually, especially in Surabaya. And Ebola also virus disease and coronavirus disease 2019 still circulate in the environment. This is a very important thing. Like this one, so many cases like tuberculosis in different countries still there and hyper and spreading to the other country. And also Indonesia also this is a very important thing to us, how to stop it on the tuberculosis spreading in the environment. So this is our job in the future. So this way One Health Network in University in Southeast Asia, IPAR, based on the situation in the world about the zoonosis disease and pandemic. Also, becoming the founding of the Sehun, like Southeast Asian One Health Network, is very important thing. Like in the Hun in Indonesia, Mihun in Malaysia, Tohun in Thailand, Phone Vietnam, Cambodia, Laos, yeah, Philippines, and Myanmar. Yeah, this is our network. So how to create our 2 minutes of the One Earth in Southeast Asian is very important in the future. And also inside in Indonesia, when collaborating center, there are many university, not only 7, but beginning or start 2015 founded in Gajah Mada University. After that, in the Udayana University and Universitas of Airlangga 2017, and then founded in Siakwala 2018, and then what is in Cendrawasih University, and the last time 2023, yeah, the collaborating center, when the collaborating center founded at IPB and at Hasanuddin University. So that means we have about 7 One Health collaborating center at the university, and also maybe and directs in different university about I think more than 100 faculty already have about the One Health collaborating center also. So one of the implementation challenge in the future, this climate change is more important issue actually. So challenge in the implementation, like the gap in implementing global regulation, for example, decentralization government, fragmented management. Indonesia actually for government has regional regulation and development plan that refer to the respective sectoral authority and development without considering synergy or interaction between sector and based on different legal principle. And then secondly, effective collaboration. This is our issue. Lack of cross-sector and cross-disciplinary collaboration in government, academia, and health profession still occur in many countries, especially lower middle income country. This is a problem. And also in the children surveillance in data system is still actually independent monitoring system. How is the data from the Israel agriculture from the, can integrate it together and dominate on the system resulting from the response across sector. So need to get financing capability, absence of the incentive arrangement has led to lack interest among countries to invent effort to prevent emerging infectious disease using the One Health approach. This is challenging in wallet implementation. So how is the future direction, for example? So communication between what is the experts, different experts, so we can solve the problem of the One Health system. How a future direction for One Health research? Like this is surveillance, for example, civil society and capacity building, public policy and regulation at the microbial system, sustainable food system, and ecological and environmental issue is become big issue. This is surveillance. What is translating the holistic definition while it integrated surveillance approach, like limited historical expertise and sector, for example, in features, limited surveillance capacity. So this one is needed. How to improve capacity building, for example. This is very important thing where we can start actually. So maybe start to the academic student, maybe coming to the official personnel in the services of institution, for example, from providing until central government. Issue what access to and quality of One Health data and information. Logistic challenging, such as lack resources, personnel, and legal basis integrated surveillance across multiple domain, environment, animal, human, and health system. and number, discipline, and need to be involved, including partner not currently routinely involved in violence. This is still from previous until now still a problem. So actually in the middle community country. Civil society capacity building. So according to the COVID-19 pandemic has proven to be communication challenging with misinformation and distracting to the result rumor spreading about incorrect information. This is a how to manage of the information give direct to the publics how to manage actually this is a very important thing with exactly data and etc. Without addressing people concern, there is a really risk that rumor will grow and is said about the COVID-19 vaccine will increase actually. So, you know, remember about the first vaccination in Indonesia about the COVID-19. The first, our president immunized using COVID-19. And then after that, more than a billion of the Indonesia people want to take up the immunization. So the public policy and regulation. This policy cover global trade, commerce, and security. How is the public policy, for example, international trade law has generally been more consistent, successful address economic issues. And there is agreement of the political attractive advantage, for example, in contrast, international health legal instrument, for example, international agreement, human, animal, or plant affect trade and wise the related to the success of the trade agreement. For example, there are financial laws associated with laws, travel business, and etc. This is, this is a reason from the time at all about the public policy. So what is antimicrobial resistance? The global health issue, antimicrobial resistance is one that the best illustrated the One Health approach actually here. So how is linked to this three component, the responsible and excessive use antimicrobial in the forest sector, like or namely agriculture, livestock, and human medicine. This storage of the aminobutyric affect global health system. Currently, it causes antimicrobial resistance like microbial difficult to treat because antimicrobial are increasing ineffective against this infection. So that's why maybe in the future, so we can thinking about the bacteriopage, for example, they can remove a point of the infectious disease and the new properties, resulting in a higher probabilities also this our problem in the future too. So sustainable food system, the ability agricultural system in high and low middle income countries to successfully feed 9 billion people by 2050 without depleting the planet finite resource and about the complex sociality. So the challenging of the appreciation and maintaining food security today, global marketing characterized and complex problem has no essay to straightforward solution and require one health approach. So like this one, this is so we can consider, we must consider about it. This is our planet. So inside there are different kind of the life, animal, plant, yeah, and ecosystem also. Ecological environment issue, ecosystem approach offer respond and the right environment and health challenging in the One Health concept provide system. However, many challenges remain ecosystem-based approach to health target. Ecosystem approach require a very different mindset from cellular and sectoral thinking, especially as human health policy increasingly focus on treatment rather than prevention. This is a ecological issue. Environment health has been reduced to simplify avoiding exposure to chemical or biological agents that are harmful in rural health. And fish occurring increases animal health. This is a lead to shift in our relationship with wild domestic animal, which are seen in the potential hazards in the human. So we go to the conceptual model. The conceptualistic linking, yeah, some government have also the concept model how to leading or to bring up the one health system good leading in the public. The concept political linking human animal environment plant agriculture and issue is not new. As so in the figure below, there are multiple factor impact health, animal, ecological, human across nation. Data is needed from various sources, effectively, strategically, determine interconnection across the domain. This is very important. There is vital role in the play One Health system in the workforce, actual problem identification and local infection. So this community on one workshop assisted by technology tool. This is important. Very important to train in communication skill, technology, application, further the education and behavior change, basic health knowledge to the they can provide grassroots linking between community and formal human health care, animal and agriculture. So every one of us will follow evidence-based protocol and report advanced practitioner, such as nurse, physician, veterinarian, and agricultural officer. This is interpreted data will be aggregated and disseminated in regular national expert. Will injure and decision-making promote how to health of the public and cheap. That means this is should be measurable outcome. It's a very important thing. So this is a conceptual one health system. The first, the means actually human animal plant environment, and this is a local. So you can build like now, and then after that, region, state, and nation, and etc. And then support to the different data. This health system, one health system. This is a good coordination, good information, good achievement, and minutes. That's why the four many kind of factor actually individual communication system and individual communication system also so expect outcome and the factor maybe biological factor socio-economy knowledge etc then incorporate knowledge yeah the expected of the outcome improve is before communities socio-culture norms industrial services and also improve competency community workers. This is a outcome. Yeah, improving communication, expedited referral, early identification, intervention cases, disease prevention, and etc. And the system policies and guidance or guideline, finance, technology, and factor and the outcome improve surveillance. This is a very important. They related between the basic and the outcome more important thing. So the conclusion, the most intention around one has focused on policy and economic problem solving at this moment. The academic group, government, agency, NGO can and should meet strategic planning around the solution complex global one and problem requirement, interdisciplinary expertise, cooperation, and creative problem solving. So, however, practically one approach needs to start community level, whereas there is strong need of the one for the unread data collection, health information dissemination. So, innovative one health approach is the potential to bring the new trained workshop equipped with health technology and rural communities to improve the health provision, animal plant, and environment. So furthermore, the benefit one health workshop are vital for surveillance and emerging diseases. So this is why academician may be a very important thing. The people how to drive of the what is the implementation of wellness in the public, like in the area chronic health problem growth of the surely by the systemic collection data, for example, can to enhance how to enhance the monitoring outcome, for example. Given the interconnectedness for all global community with human, animal, plant, and environment impact each other and do not respect geopolitical wonders. We believe it's time for country consider training community-based one health workshops. Thank you. very much for your attention. This is our presentation. Thank you. Assalamualaikum warahmatullahi wabarakatuh. I give to the moderator.
Okay, thank you, Professor Fedik, for your interesting topics. Now we come to the Q&A sessions. We have around 5 minutes for these sessions. Let me check in the chat box. I think we have one question from one of our students. The questions will be, how does the One Health approach enhance our ability to predict, prevent, and control zoonotic diseases? This one, the question from
Yeah.
our participant, Prof.
Yeah, thank you for the question. This is a very important thing and good question. Actually, the first should be improve yourself. Actually, how to detect of the cases, detect of the infectious disease in the field, for example. So that's why you must improving the technology in the laboratory. For example, a different method you can improve by yourself and can to detect very easy in the field using higher technology. And then you can characterize of the infectious disease, which one actually what is the agent, for example, still feeding in the environment, for example. This is very important thing. So one handbook for student, I think very important. So that's why in this is cases we have programs, special One Health Collaborative Center for student. Yeah, for student. This is routinely every year. You can take participate in One Health and our program, One Health Collaborative Center at the BMC Erlangga University. But and abstractly, how to use the technology from the laboratorium, and then you can bring information that to the field. This is our idea. Yeah. Thank you. Sorry.
Okay, thank you very much, Prof. Fedik.
Yeah.
I think we still have another questions from our alumni, I think from Dr. Saifur, but I couldn't read it clearly. Wait for a moment. For the technician, could you please make the questions bigger so I can read it easily? Or before that, I think we have one more questions from our student as well, Prof. Before
Okay.
we are going to the Dr. Saifur's questions, uh, the, qu- the question is, what are the key challenges and opportunities in implementing One Health strategies in veterinary practice, particularly in resource limited setting? So this
Yeah.
one's the question.
So the big issue, I think, based on the social economy and the what the farming, this is very difficult to manage because the farming in Indonesia is not all right man or right farming now, but small farming is dominate in our country actually. And the secondly, and the issue of the one let's implemented I think between between association very difficult to meet and together discuss how to make a problem solving with in the future one health problem actually like antimicrobial resistance, for example, how to manage actually to stop it of the spreading cases or agent like. Food and mood disease, for example, how to minimize and to localize is very difficult. This is the regulation in Indonesia, I think, still centralized. Although in the regional also have special special action, but still the decision depend on the central government. This is our problem. And secondly, how to split or to dissemination about our decision from the central government to the regional government and to the farm, for example, to the public, very difficult. You know that in Indonesia, so many kind of ethnics. So that's why needed and the attitude, for example, ethic, bring to the approach. What is this is one system to implement in the public. This is still, yeah, I think still a problem. Although in the future maybe we can create some kind of like regulation, for example, more than very easy, simply to implement it to the public. Yeah, thank you.
Okay, thank you, Prophetic. Actually, we still have several questions, but unfortunately, due to the time limitations, so
Okay.
we will collect all the questions and we will put in a one form and give to the Professor Fedic, and
Okay.
he will reply, and we will give the feedback to all of the audience from
Okay.
the
Okay.
questions. I think that's all, Prof, for
Thank
the
you.
first sessions. Very nice and very uh, impactful presentations. Thank you, Prof. Eddie.
Welcome.
Yeah, Prof, please stay with us because now we move to the certificate awarding. Okay, so
Okay.
before we end the session from the first speaker, we would like to give a certificate of appreciation to Prof. Eddie. Prof, we would like to give the certificate of appreciation. Please receive the
Okay.
certification. Please receive the certificate of appreciation. And once again, thank you very much for sharing the knowledge with us. Thank you, Prof.
Okay.
Yeah, see you
Yeah,
in the next
thank you very
event.
much for all organizing committee. Yeah. Okay, thank you. Thank you. See you next time.
See
Yeah.
you, Prof.
Okay.
Okay, now let's continue to the second presentation from our second speaker. Our second speaker is Professor Edogan Memili, DVM, Master of Science, PhD. And in these sessions, he will deliver the topic entitled Food, Animal Health and Production for Global Food Security. Before he starts, I would like to introduce our second speaker. Currently, Professor Edogan is a professor and an executive associate director, College of Agriculture, Food and Natural Resources, Prairie View, A&M University, United States of America. For academic background, he graduated and got his Doctor of Veterinary Medicine from Veterinary Medicine Istanbul University. Then he continued his Master of Science at the University of Wisconsin-Madison, majoring in bacteriology, and he pursued his Doctor of Philosophy, majoring in endocrinology and reproductive physiology in Wisconsin-Madison, and followed by a postdoctoral in genetic and epigenetic at Harvard Medical School and sabbatical on genetic at the Cornell University. His research interests are systems biology, functional genomics, epigenetic, embryo, chromatin structures, assisted reproductive technology, DNA methylations, semen analysis, gene regulations, and also stem cells. He also active in producing many international scientific papers in related fields and has published more than hundreds of scientific papers, and he has a Scopus, a 33 age index Scopus. Ladies and gentlemen, please welcome Professor Edogan Memili. Hello, Prof. Are you with us now?
Yes, hello everyone.
Hello,
Thank you very
Professor.
much. Can you hear me well?
Okay, yeah, we can hear it very clear.
Very good. I wanted to thank you all for attending. I want to thank director. I would like to thank Prof. Mirni Lamid, Dr. Zulfi Rasyada, and all of you to make this thing happen. happen. So my talk is going to be a little bit different because I would like to share with you, let me minimize this, food animal health in the context of improving their reproductive efficiency. I appreciate that a nice introduction where I also want to mention that science is exciting. The best is yet to come. So with all of the students in the audience, there's a lot of work to be done. Currently, I serve as the Associate Dean for Research, as well as the Director of the university's research farm on Prairie View A&M University campus, which is located half an hour north of Houston and half an hour south of College Station in Texas, United States. You see the map over here, the US. Texas is one of the largest states in the country, and the university is located over here. It is a land grant HBCU, or historically Black college and university. So in the university, we have the College of Agriculture, Food and Natural Resources. And because it's a land grant university, we do research, teaching, and extension outreach. In the research, I oversee the Cooperative Agricultural Research Center, and there are 6 domains of excellence in the animal systems, plant systems, food and nutrition systems, social systems and allied research, natural resources and environmental systems. And recently we added artificial intelligence. In the farm that I oversee, we have a lot of centers, including International Goat Research Center, Meat Science Center, Poultry Center, the greenhouses, horticulture, and recently algae biotechnology. If you are interested in, I encourage you to go to this webpage and learn more about our research programs, especially pay attention to the International Goat Research Center. As you see, the first word is international. So let me briefly tell you a little bit about the land grant system. Ladies and gentlemen, this is a very unique one because at this university, we are required to do research, quality research, innovative, transformative, translational. And the research project must have the best technologies, experimental design, and it must be significant. That is, it must produce new knowledge and some kind of application to solve a problem. The researchers, like professors, must be active researchers who are doing quality research, attaining grants, publishing peer-reviewed articles, and presenting. We also have to have a quality environment to do research. That includes the equipment, facilities, and all that. In the teaching mission of the land grant program, we must teach students through experiential learning, learn by doing. That is, they go to the laboratory, they do things, they learn. That is evidence-based. We have courses with laboratories, so they learn the theory, and then they learn the practice by actually doing. And this extension is an American thing that's not very common abroad, but it is very, very helpful because extension must take the knowledge that is generated at the university to producers, to stakeholders, to meet their needs. So as you see, we have the research, teaching, and extension outreach. I direct the research part of the faculty's program, but I also work closely with teaching and extension programs. In the research, we have the system science with all of the 6 areas. Resources, our goals are to do research, education, extension, and ultimately make an impact on agricultural science and society, not just in Texas, not just in the US, but in the entire world. Ladies and gentlemen, we have a major problem that global population, human population in the world is facing. That is food security. What do I mean by that is that everyone on earth must have available food for them. The food must be affordable. They can pay for it. And the food must be accessible. They can go get it. So these 3 factors I define as food security. And the world population is expected to be over 9 billion by the year 2050. What that means is that we have to produce healthy food animals, such as cattle and goats that I'm going to talk about today. But remember, I mentioned healthy because health is the number 1 requirement, healthy animal. So there are some problems and opportunities. For example, because of the increased human population, there's a huge need for proteins, such as meat and milk, from animals that the new growing population of humans need. There are emerging diseases. Prof. Rantam did a good job. I appreciate you for talking about One Health, very important. And with the climate change, increased temperatures in the world, and increased technological advancement, there are a lot of emerging diseases. Therefore, we have to have healthy animals. The lands for animal agriculture are shrinking. That means we have less land to produce animals, crops, plants, and all that. Opportunities, there are a lot of them. All of the students in this audience, in my opinion, are opportunities because they are going to help us solve these future problems. There are a lot of research areas, including animal health, nutrition, genetics, as well as the way we produce animals, precision livestock, systems analysis of the whole food animal production that must incorporate artificial intelligence. Smart agriculture systems we must incorporate. So that means One Health is the center of this whole food animal production. In the United States, we at the university make sure that our animals have these 5 freedoms. They must not be hungry or thirsty. They must not have pain, distress, discomfort, and they must be able to behave naturally. We use a lot of animals in our research programs, and we are required to have the approval of the Institutional Animal Care and Use Committee. Center for Disease Prevention and Control has a nice One Health concept information over here. I encourage you to read more about that because animal health is critical for human health, and that we got to have healthy environment that has the soil, water, air, plants, and everything. So folks, we got to have this One Health before we can produce food, before we can produce research and everything. So as far as the science of production of food animals, there are also problems. I see them as opportunities. because we have to produce livestock such as cattle and goats with this new transdisciplinary science and cutting edge technologies. We must have genetic evaluations, testing the animals, we call it progeny testing, and we must have really good phenotypic data. For example, how much milk is being produced, how much meat is being produced, the quality, and then we must take those, associate those with the genotypic information so that we can select the best animal. We have to select the animals using breeding soundness exam. More importantly, in this age, we must incorporate artificial intelligence. In our laboratory, for example, we are doing AI or AI. We are doing artificial intelligence in artificial insemination of livestock. And we need to produce animals much faster, much quicker through precision farming of food animals. As for the research that is in the food animal production, there are specific gaps in the knowledge base and technology base. For example, we need potent biomarkers, in this case, to evaluate semen quality and predict male fertility of bulls, bucks, and all the male animals. Because a lot of farms are using artificial insemination. That means sperm from one bull, did you know, can be used to inseminate millions of cattle. In fact, there is one bull called Starbucks, nothing to do with the coffee shop. That guy has over 1 million children. So that means we have to choose the bull very, very well. We have to have animals with phenotypically distinct characteristics so we can identify markers. We must do at our universities both discovery studies and mechanistic studies that have to be robust, repeatable, with the right vigor. In this day and age, ladies and gentlemen, science is being transformed. Rather than looking at millions of cells all in one tube, now the sciences has advanced to single cell analysis, microscopy, where we can look at the cells, proteins, as far as locations, the levels, and all that. And we must incorporate molecular and cellular anatomy and physiology along with biochemical analysis to see the full picture of a developing animal, from bull, for example, early days until the end of its life. To understand the full picture, we must continue cutting-edge research. Information is power. Years ago, my graduate student and I have uncovered the world community together, the genome of the cattle. Briefly, I will talk about food animal production from a point of fertility. As you know, there are lots of bulls, in this case, dairy Holstein bull and Angus meat breed, beef breed, but their fertility differs like this. Low fertility animals, high fertility animals, and a lot of them are average fertility. We want this group of animals here that also have other genetics. Do you know what happens to these animals here that are very low fertile? And if those animals do not have good genetics, they become hamburgers very fast, don't they? Say yes, please. So genetic, epigenetic, environmental factors all affect bull fertility. And if we have low fertility, that means it's a huge economic loss. Early on, I studied with my students and collaborators how early embryos develop. At the beginning of life, with the fertilization, embryo starts developing, activates its genome, and when it gets to blastocyst stage, we transfer into a recipient, a cow, After 9 months, we have a miracle animal on the ground. So we would like to understand the role of the sperm driving embryonic development. Now, egg has a lot of proteins too, and RNA, DNA as well. So we tested the hypothesis that sperm brings more than half of the genome. So let me share with you briefly about ways that we study increasing reproductive efficiency and production of food animals, primarily the cattle in this case. My students, collaborators, and I, we wanted to test the hypothesis that sperm from these low fertility bulls have molecular and cellular defects that conventional techniques cannot detect. But these defects prevent these animals from having high fertility. So we always start with an experimental design that is so robust with statistical power in place. In this case, we take samples from bulls with different fertility, selected out of 1,000 or more bulls, and we do omics studies, proteomics, transcriptomics, metabolomics, lipidomics, and some of the reductionist techniques like real-time PCR, quantitative, microscopy, and we use computational biology, bioinformatics. For example, to understand what some of these protein markers are interacting with, we call them interactome. And in some cases, we do in vitro fertilization to understand the mechanism. Just a couple of examples. For example, we showed protein differences between sperm from high fertility versus low fertility animals and showed a lot of proteins with important functions differ between the low and high fertility. Over the years, these have been published. Out of these 125 proteins we identified many years ago, some of them have been now used by companies as biomarkers to predict male fertility of not just animals, but of livestock, but also other animals, endangered species, as well as human men as well. So let me tell you a brief story about the sperm nuclear protein called protamine. These proteins are in the center of the DNA, which is wrapped around this protein so that it is very tight over here. This cell is only about 2 to 3 micrometer in diameter, and it has to be so compact, so tightly packaged, so that the DNA can be protected from damages like reactive oxygen species, and the sperm head can be so compact to move ahead. To do that, we got animal sperm from high and low fertility, and they have been used over anywhere from around 550 to 11,000 times. And we did flow cytometry, nuclear shape analysis, immunocytochemistry through the experimental design and materials and methods. My student, Dr. Shula Doan, during her PhD, showed that sperm from low fertility bulls have much lower of this protamine 1 compared to high fertility. Also, sperm from low fertility animals have a lot more DNA damage, and then they also have a lot more membrane damage. This was published years ago in Biology of Reproduction, one of the best journals in the field, and it has still been used, and it is still one of the most potent markers for fertility. Students make a big difference because over the years, they studied sperm RNA and showed that there are specific microRNAs associated with low or high fertility. Just to save time, I want to show you some of their work over the years, the RNA work, mRNA, total RNA, microRNAs in different studies, as well as metabolites in the sperm and the seminal plasma of food animals, as well as lipids in these sperm cells are all associated with fertility of food animals. So we also looked at the epigenetics, factors that are beyond the genetics, and our students, led by Dr. Nasir Kuchi, showed that these proteins important for fertility have different acetylation or methylation, and that their modifications, we call these post-translation modification, are very important for the fertility. Because depending on acetylation or methylation, these proteins can either suppress or activate gene expression at the molecular level. So as for the bull part of the project, we conclude that in order to evaluate semen quality and predict bull fertility, we have to have an integrated approach that has to evaluate the genetic of the animal and look at all the gene expression. I showed some examples, and other tests, including computer-assisted sperm analysis, flow cytometry, biochemistry, and all of that, so that we identify the most fertile animal. Folks, last part of my presentation has to do with the goats. Here at Prairie View A&M University in Texas, we have the International Goat Research Center, where we have 2 projects. And we're asking the question, can we use the knowledge gained from bull for understanding buck sperm? Specifically, we have 2 projects. One is to identify biomarkers for fertility, including biomarkers of thermotolerance. What I mean is that, what are the biomarkers in the animals, in this case sperm, for climate resiliency? How can these animals withstand increasing heat? We also want to develop extenders for improved cryopreservation of the cells. Very briefly, we know that all the ruminants are clustered around here. They are similar, bulls and goats, and that they are both ruminants. In the world, there are close to 2 million goats, actually 2 billion, and there's about 1 million goat population in the state of Texas in USA. The largest goat population in the US is in Texas. There are different breeds for both meat and milk, and there's increased demand for goats because I saw in Indonesia, you have kacang goats that kind of climb up the mountains, but they need to be improved, milk production, meat production, and all that. In Texas, there are a lot of international populations, people from all around the world. They want goat meat, goat milk, goat ice cream, goat yogurt, goat anything. So we would like to look at the goat, and when we look at organismal level, they are quite similar, except size is different between bull, which is much bigger, much larger than the buck. When we look at the molecular level, they have different chromosomes, and that their genome sizes are different. So if you look at the molecular basis, they have some differences coming up. If you look at the sperm itself, molecular anatomy, they're quite similar, but sizes are different. If we look at the structural characteristics of the sperm, as far as mitochondria, plasma membrane, they are similar, but not the same. That means there are differences in the protein content, phospholipids, and the cholesterol that are so important in order for us to develop extenders so that these cells can freeze better. Another characteristics as far as the sperm are somewhat similar, but there are differences exist. I wanted to share this with you because studies in the goats must be repeated because they are not strong and published in literature, or they have to be much additional studies needed. So, folks, we looked at the literature, identified genes that are important, in this case, DNA. There are single nucleotide polymorphic mutations in some of the genes with specific functions. For example, this Izuma1, this is very critical in sperm and egg fusion. If there's a mutation there, the sperm and egg cannot fuse. Likewise, we found a lot of important proteins, including those reported from our laboratory, that these proteins are very important for either at fertilization or after fertilization, because sperm has the function of fertilizing, activating the egg, and supporting embryo development. So when we look at the molecular similarities of these genes, in this case proteins, amino acid sequences of all of these biomarkers between goats and cattle, we found very high level of similarity, almost 100%. So we hypothesized that structural similarity, in this case amino acid sequence, tell us that they are physiologically similar too. That is, their functions are similar as well.
Sorry for interrupting,
We found
Professor.
RNA markers. Yeah, I'm
Sorry.
finishing up. Thank you.
Sorry, your
That
time
they are
is
different.
5 minutes left.
Yes. Thank you. So RNA markers are across the sperm as well. As I mentioned, that protamine is a very important one. In fact, there are 3 of them with critical functions in the sperm head. If we look at the molecular sequence using bioinformatics, we see there are a lot of conservation, and there are a lot of motifs too. These conserved amino acid domains that tell us there are significant functional sort of motifs. So, folks, as I wrap it up here, bull and buck sperm share a lot of similarities. At the molecular level, they are similar, but there are differences as well. The buck is not a bull. So studying both of the organisms will help us advance food animal production, reproduction specifically. And that moving forward, we have a lot of research opportunities to do using advanced technologies, whole genome sequencing, single cell analysis, much more comprehensive studies, and putting all these together with collaborations across the world to generate new knowledge. I would like to thank the United States Department of Agriculture's National Institute of Food and Agriculture for the competitive grant, as well as Evans Allen grant. And I have a postdoctoral fellow from Turkey who is funded by the Scientific and Technological Research Institute of Turkey. I would like to thank you all for your attention, and I would love to have some questions. Thank you.
Okay, thank you very much, Prof. Memili. Now we move to the Q&A session. Actually, there are several questions that has been sent to us, but due to the limited time, so we will limit the questions, into 2 questions to be read, but do not worry because the rest of the question will be collected and sent to Prof. Memili to be answered. Is it okay, Prof?
Yes.
Okay, thank you. So I would like to read first question. This is from our final year students. So the question is, what are the main challenges in ensuring the health of food animals that directly impact global food security? And how can these challenges be effectively addressed?
Yes, so we want to make sure that animal health is number 1, in that we have to prevent diseases from happening. That means we have to vaccinate all the animals with the available vaccines. We have to monitor the animals to make sure that there are no diseases coming from wild birds and others. As Prof. Rantam mentioned, disease like bird flu and everything are very important. So, vaccination, monitoring, and providing the best care for animals of those, especially the five freedoms, are very important for us to keep animals healthy. There's a big concern right now because of the climate change and technological improvements around the world, or buildings coming up where animals don't have enough space. There's air pollution. So all of these things affect animals just like they affect our health. So we have to be more mindful about One Health to ensure that we are healthy humans. Remember, there are zoonotic diseases that go from animals to humans, from humans to animals, and our environment has to be clean, and that includes the water, the air, and plants must be healthy as well. So overall, healthy world, healthy everybody. Thank you. Any other questions?
Yes, actually we have,
Oh, let me Yes, let
yeah.
me address buffalo biomarkers. They require phenotypic data collection that, for example, milk production, meat production, growth and development, all the economical traits must be measured. And then we need to get the genomic sequence of these animals so that we can make the associations between the genomic information and the phenotypic data to identify markers.
Okay. Okay, thank you, Prof. And this is another question from Toulouse. The question is, how about development of buffalo molecular research in US, and what about difficulty in buffalo for biomarker discovery study?
Yes, so there are, right now the science is advancing so much, the new technologies, artificial intelligence, very, very sophisticated equipment coming up with microscopy, gene sequence, all of those things are coming down to very, very precise, minute level. So we must incorporate those in our research. But first and always is the fundamental, the best question. And then with the best technologies, we must address. With regard to the buffalo, I saw buffaloes in Indonesia and in other countries as well, and that we must identify or collect phenotypic data that are economically important, how fast they grow, how much milk and meat they produce. And then we need to get the genomic sequence and make associations between genomic sequence and the phenotypic data to get those biomarkers that can be used in precision breeding and selection.
Okay, thank you, Prof. This is last question that I would like to read. Okay, what a great person, oh, sorry, this is from, no name. Okay, so he said that what a great presentation, Prof. Memeli. Thank you for inspiring us about reproductive technology. So we are from Artificial Insemination Center, Faculty of Veterinary Medicine. We have some question. First one, what kind of extender that you developing now? So the first question is what kind of extender that you developing now? And the second one is how about sexing sperm in sperm bag or bull in there?
Very good, thank you. We are studying the cell, bug sperm cell, to develop the extender that will suitable for that cell. What I mean is that when we use the bull sperm extender for bug sperm, it doesn't work well. Why not? Because we hypothesize that these cells are different. For example, there are different lipid levels, there are different phospholipid contents. Actually, their phospholipid membranes, the cell membranes, are different. So we are studying how those membranes are, and based on that, starting with simple salt, buffer, milk versus egg yolk, we are using all of these different protein contents to develop effective extenders for bug sperm. Meaning, we would like to freeze the sperm in these cells, in these extenders, and thaw so that their post-thaw will be very effective. So we are still studying those by simple buffers that contains the salts, antioxidants, cryo-damage inhibitors, as well as proteins, either milk or egg yolk. Now, with regard to sex sorting, the separation of X or Y bearing chromosome of bull sperm is very common. There's a company called near Prairie View A&M University. It's called Sexing Technology or ST Genetics, Sexing Technology Genetics Company. That's what they do. They use flow cytometry to sort the sperm because that's very important for producers. For example, for the dairy producers, they don't want bulls. Bulls cannot give milk. So as a result, it's becoming very common. However, the pregnancy rates as well as the cryopreservation efficiency of sorted sperm is much lower as compared to the fresh or regular unsorted sperm. And for the buck sperm, those are not quite widespread. But we eventually would like to do that, but the companies are not doing that as much as they do bull sperm. I appreciate all of these questions. Great questions.
Yeah, thank you very much, Prof. Unfortunately, due to the limited time, so we have to end the Q&A session. But do not worry, all the questions will be collected and sent to Prof. Memeli. Yeah, so...
Yes, I'll be happy to answer. Time flies by when you are having great time. Thank you all.
Thank you so much, Prof. That's all for the Q&A session.
Okay, before we end the session from the second speaker, we would like to give a certificate of appreciation to Professor Edogan Memili. And
Terima kasih.
Okay, once again, thank you for sharing your knowledge with us. Thank you, Professor.
Thank you so much, Prof.
Thank you all. My best wishes to everyone. Have a good day.
Have a good day too. Okay, now let's continue to the last lecture of the keynote speaker session from our third speaker. So our third speaker is Dr. Aksorn Saintin Chai. She will deliver the topic entitled Pharmacokinetics and Toxicokinetics, Implication for Drug Development and Safety Assessment. Before she starts her presentation, let me introduce Dr. Aksorn first. So currently, Dr. Aksorn is an assistant professor in Department of Pharmacology, Faculty of Veterinary Medicine, Kasetsart, University, Thailand. For academic background, she graduated and got her Doctor of Veterinary Medicine from Kasetsart, University, Thailand. Then she pursued his Doctor of Philosophy majoring in Environmental Toxicology in Hokkaido University, Japan. Her research interests are pharmacokinetic and toxicokinetic, xenobiotic metabolism and environmental toxicology, molecular technology in pesticide toxicology. And she has published 23 international scientific papers and 3 national publications with Scopus Index 11H Index. Okay, ladies and gentlemen, please welcome to Dr. Aksorn. Hello,
Okay.
Dr. Aksorn.
Hello.
How are you?
So let me check my voice. Is
Yeah,
clear?
we can hear you
Okay.
clearly.
So should I share my slide by myself or you
Sawadika,
prefer
Doctor.
to? Sawadika, sawadika, sawadika.
Sawadika. Actually, I only know one word, only Sawadika.
Okay.
Okay, so are you ready to start your presentation?
Yeah, of course.
Yeah.
But I like to know that should I share my slide by myself or
Yeah.
your?
Yeah, we
Yeah,
can help
okay.
you to share your slide. We can
Okay,
help you
okay.
if you want, if you
Okay,
don't mind.
let me share my slide first.
Oh, you would like to share by your own? Okay, okay.
If possible. Okay.
Yeah,
Can you
Doctor,
see?
your presentation time is 35 minutes. Thank you.
Okay.
Yeah, the time is yours.
Okay, so first of all, I would like to thank Kai introduced me. Really excited, and also good morning, all delegate and participants. This is first time that I joined this event, and I like to thank the committee of the fourth ICVMSS 2024 to giving me a great opportunity to be here today. I would like to share the topic of pharmacokinetics and toxicokinetics in terms of application for drugs development and safety assessment. So nowadays, pharmaceutical company, okay, companies in both human and animal segment dominate the global drugs discover market. The trend of global drugs development is significantly driven by the rising prevalence of chronic disease that we know already, and the growing of the uh, genetic population. It means we have the long life, and our animal also long life also. So it may create the huge demand for various innovative drugs and increase demand for the drug across the globe has through the drugs discovery market. So uh, to produce the new drugs or drugs development, we cannot ignore the basic of pharmacology and toxicology. Toxicology to prove the drug products to be safe for patient and also our owner also. So I would like to remind of the background of general pharmacokinetics together with toxicokinetics. Then we will go to understand the drug products performance belong to biopharmaceuticals and some testing results such as bioavailability and bioequivalent. Finally, the drug products quality also should be concerned. So generally, in terms of pharmacokinetics and pharmacodynamics, belong to how body affect to drugs and how drugs affect bodies. So we know that a drug is mainly absorbed into the body by oral inhalation and injection. Then that is distributed to blood circulation, target organ, and change uh, form under biotransformation via phase 1 oxidation and also phase 2 conjugation before increase into urine and feces as the main. During drug distribution in target organ, drug effect occur from the receptor binding, signal transduction, and physiological effects. Within pharmacokinetics and pharmacodynamics, kinetic parameters such as the elimination half-life, as we know as well, plasma coherence, the volume of distribution, Cmax, Tmax, and area under the curve are one of the criteria for drug development. Moreover, a drug interaction is also concerned for drug development since it may affect to biotransformation, such as the enzyme induction or enzyme depression. Since in the veterinary field, we have various factors concerning when using drugs, such as dosing, route of drug administration, interval, and adverse effect or side effect. Meanwhile, we should consider the toxicity of drug as well. So, to concern the toxicokinetic and pharmacodynamics, the main concept is look like the pharmacokinetics, but we are just focusing on the toxicity of the chemical or drugs that we use because we like to know about the effect and responsibility that will be occur in the body, in our pet body. So there are many factors determine toxicity. For example, exposure, dose, intrinsic toxicity of substance, and intrinsic response of the target. So in case of drug toxicity, term of lethal dose, maybe you know LD50, lethal concentration, no observed effect level, no observed adverse effect level, lowest observed effect level, lowest observed effect levels. If this kind of the parameter is maybe concern in the drug development also, especially the LD50. So, as I mentioned, the kinetic parameters are one criteria for drug development. So, to understand the process of drug development, this slide shows the cycle of drug development as well. So, in the drug development market, there has maybe they try to build up the new one, the new drugs, like the original one, or they like to build up the generic drugs. What is the generic drugs? Generic drug is the some process or drugs with the same active ingredient as the original drugs and work in the same pathway but may look different and contain different non-active ingredient. So should be they have to be the some method to prove the generic drug is similar or almost similar as the original drugs. And one thing that about the generic drugs are often cheaper than original drugs. Then, in order to start the drug development, the scientists or researcher or the pharmacologists try to start to research the chemical, screen the chemical in environment or in plant or in some microbials. And then they're looking for the chemical that can affect, then evaluation the candidate drugs in vivo and in vitro study under approval laboratory condition. This is very important. Then after they got some information or some result in the preclinical development, they move to study in the target species. So to evaluate the clinical safety and efficacy in the target species that I said. So this process, we have to concentrate on the species difference and specific species also. So in that term, the pharmacokinetics and pharmacodynamics or toxicokinetic and toxicodynamic data will be important to submit the regulatory authority, and products will be approved or not. It depends on this result. After they get some approved, so they can start to do the manufacturing, marketing, and also on our hand to use in our animals, our patients. So in laboratory, in the laboratory, candidate drugs or active ingredients are not usually given as the pure chemical drugs substance, but they have to perform like a formulate or dosage form before using. So products performance is coming in this process because the drug product performance is performed to study releasing drug substance from drugs product, either for local drug action or for drug absorption into the plasma for systemic therapeutic activity, and also focus on developing quality drugs products that are safer, more efficient, more effective, and more convenient for patients. So generally, the drug formula is totally belong to the application, right? Like a systemic or local therapeutic treatment, the quality of drug form, like a solid or liquid or emulsion. And fairly is fairly used or not because it is go toward for the marketing, right? And finally, this product performance should be achieve the specific therapeutics objectives. So in order to achieve this, the candidate drug product should treat the acceptable rate of drug release here from starting here, along with absorption, distribution, and elimination at will. Not only drug production performance, drug product quality, as I show here, also the one defect which may injure or death in the target organ. So it means that we have to looking for the good quality protocol or good quality method to prove that the drug development of our new drugs to be good for use and stability and sustainability. So to apply the concept of the pharmacology and toxicology in drug development, biopharmaceutical intend to adjust delivery of the drugs from drug products in such a manner as to provide optimal therapeutic activity and safety for the patients. And the interrelationship of the physical, chemical property of the drugs and dosing form in which the drug is given, and the route of elimination on the rate and extent of systemic drug absorption will be useful for the drug development and drug production. Like in this figure, this is the proof of the oral biopharmaceutic. So before approval, we have to study about the solubility, dissolubility rate, and some absorption rate, pharmacokinetics, and pharmacodynamics, and also some technology for the drug delivery. So consider biopharmaceutics in drug products design. We consider parameters which belong to pharmacodynamics. For example, therapeutics subject. We should define the therapeutic index, the range of the safety, right, of the candidate drugs together with toxic and adverse effects. And we don't forget about the interspecies difference, since this is an important point that we're different from human. And treatment in veterinary is also different from human treatment. On the other hand, the chemical and physical property of the drug product, pharmacokinetics, bioavailability, route of drug administration, decide drug dosage form, decide dose of drugs is also considered. Moreover, it is also important thing when you start to produce some drug development, you have to concern about the cost and stability and also quality control together. If the drug is very high effective, but the cost is too high, so sometimes it's not good in the real life for the using. Okay, so let's start considering the products consideration in the solid form. This is the usual and commonly that the pharmaceutical company produce in the solid formula, like a tablet and a capsule. If we know the characteristic of the candidate drugs products, we can estimate the rate limiting step after drugs product show the integration and drug release. And in drug development, the limitation step of the lipophilic and hydrophilic drugs help us to be concerned how drug accumulation in the body and the possibility to toxic when applied to animals. This is sometimes that the pharmaceutical company concern, and sometimes they use the lipophilic drugs because they need some effect in the target organ, or they need to protect the active drugs before go to the target organ. So it depends. So, moreover, various physical chemicals property also include when producing new drugs. For example, the drug product has proper pKa, pH, polymorphism, that which are necessary for the stability and solubility. Another thing, the functional coefficients present the relative affinity of the drugs for oil and water, as drugs that has a high affinity for oil may have poor release and dissolution from the products. Another thing that may be also that not maybe that's important. This is the excipient. Excipient interaction may affect the stability that one of the important in the producing drug products that I will explain in the next slide. So in the formulation factor affect products performance, one thing is yes, the one that can I continue? Okay, so the one thing that
you still have time,
may
doctor. Yeah, you still have time.
Yes.
Yeah, you can continue your presentation. Thank you.
Okay, and the one thing that are important is the excipient. In our experience, maybe we know about the vehicle. The vehicle is a subset of the excipient. Excipient in the drugs product may also affect the dissolution kinetic of drugs, and sometimes the excipient can affect together with the drug and support the drugs. So in the table, show the common chemical that we use for the oral liquid drugs products and also solid drugs. For example, you may know about the sorbitol, alcohol, propylene glycols. This is propylene used in the dosage form for the sweetie, for the preservative, and solubility. And also in the solid drugs, you may know about the lactose, sucrose, methylcellulose, cellulose acetate, and these are common for the using to produce the dosage form, like in the capsule, encapsule or coating the tablet form. So it means that maybe excipient will be effect to the drugs. So this study show the pharmacokinetic parameters on the oral products for the excipient. Regarding the previous slide, we know that the cellulose acetate tablet is the entering code for the to make the tablet or capsule. But in the kinetic parameter, they show the decreasing of the decreasing of the rate constant, and for the Tmax is increasing, and for the curve is decreasing or no effect. So sometimes we should consider about the vehicle or excipient also. Moreover, the effect of the drugs also depend on transporter. Where is the transporter? A transporter is the one thing that belongs to the pharmacodynamics. They have some transporter to attach the drugs before show the effective. The transporter may be support like suppress or depress our drugs also. As we know, our body, if we become old or we have some chronic disease, maybe our transporter should be depressed. And if we receive some chemical like synbiotics, maybe it's affect to the transporter or to be decrease the kinetics or decrease the dynamics effects. Okay, so I'd like to just give you the example for the important transporter that we know. First, drug-drug interaction. The drug-drug interaction is sometimes we cannot give only one drug to the animal, right? We have to give more than one, and we have to concern about the interaction between drug and drugs, and sometimes drug and diet, and also drug with some food, right? And okay, so in this concept, maybe they cause some the increased action of drugs, decrease action of drugs, or cause adverse effect. For example, we know about warfarin. Warfarin sometimes we use for the heart disease, right? And if we use the warfarin, we have to concern about the vitamin K cycle. Yeah. We have to consider and focusing on the vitamin K level in the blood because warfarin interrupt the vitamin K cycle. On the other hand, maybe we know about the pig protein. They have the multi-drug resistant protein in some species, in some dog species. So we should concern when we produce the new drugs or the products. And meanwhile, the clinical trial or in vivo study should be concerned a lot when we like to prove the new drugs or drug development. So one thing that I already mentioned before, the interspecies difference. In our knowledge, we learn a lot about the interspecies difference, about cat is not small dog, as we know well. So because of the cat or affinity family, they have some pseudogene or deficient of UGT or glucuronidase in their body. So it means that they cannot working well to metabolize some drugs, for example, acetaminophen. So we should avoid to apply these drugs to the animal because it's too toxic. But not only the affinity family, nowadays they have a lot of the exotic pet in clinic, in the hospital, and also maybe in your hand. So you have to concern to use some drugs also. For example, in this study, it's still in the acetaminophen study. So not only the Felidae or the feline family, the ferrets also should be considered when you use some drug that contain acetaminophen. So it means that species difference like metabolism or morpholism in cytochrome P450, UGT, and sulfates is also important when you like to do the clinical trial. Okay, next is the range of the safety zone, ICS safety zone, because this is the therapeutic effect to be confirmed the safety of drugs product. The therapeutic range or therapeutic window is one parameter used to demonstrate and estimate how drugs product safety or not. If you plot the graph between time and the plasma concentration, you can see the duration of action, and the upper is the side effect will occur. So there between this section, you can see the window. This is therapeutic window. But if you plot graph between the concentration of drugs in plasma and the responding, the percent of responding, you can see the efficacy together with the toxicity. This is the range of the therapeutic window. This is range of therapeutic window, but we can equal the therapeutic index as well. So how? Therapeutic index is the gap between minimal therapeutic effects, and we can use the ratio between TD 50 and ED 50 to estimate the therapeutic index. And also, you can use the LD 50 divided by the ED 50 to know the therapeutic index. And how we estimate to spot the safety drugs. So therapeutic index, the safety should be equal to or more than 2, indicate the drug is safe. But if less than 2, you have to concern this drug is unsafe. Or you can see the narrow of the therapeutic index as shown in the next slide. In this slide, I put the example of the warfarin together with the penicillin. As you know, warfarin is very high toxic and interrupt with the case cycle, and penicillin is one of antibiotics that we usually use to inhibit or kill the bacteria. So, between the warfarin and penicillin, you can see the therapeutic window is different. In warfarin, they show the narrow of the therapeutic window when compared to the penicillin. This is one thing that we can estimate that warfarin is high toxic, and we should concern when we use warfarin or when we give some chemical look like warfarin to the drugs. Okay, let's move to another parameter that is also commonly used to assessment of drugs product performance. This is the bioavailability and bioequivalent that will be used for the in vivo study. So for the bioavailability is one aspect of drugs product quality that link the in vivo. I say in vivo, right? Yeah, should be done in vivo performance of the new drugs product to original formulation. So we can estimate the clinical safety and efficacy of the drugs. So we can know as a percent, and we use the area under the curve to calculate the bioavailability. But for the bioequivalent, The bioequivalent is a drugs product performance test that compare. Now they compare the bioavailability of the same active pharmaceutical ingredient from the one product as a test to the reference as a second drugs product. So you can this is very popular for testing the generic drugs. So let me share the factor a bit. The factor influence the bioequivalent and impacting drugs development
sorry
are
for
similar to
interrupting.
the biopharmaceutical, including the product stability
Doctor,
that
sorry
show
for
the
interrupting.
yes.
Your time is 3 minutes left. Thank you.
Okay, so you can consider about the stability and the sovereignty that is useful for concern about the shelf life or the expiry date. And for the bioequivalent study in the new drug development, you have to consider about the active ingredient first, and then you can do the clinical efficiency, uh, as I mentioned before, kinetics, blah, 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 blah. And then you can get the formula that they're ready to prove and use in animal. But for the generic drugs development, we use the bioequivalent also, but we compare. We compare with the brand name or original one, and we use the parameter. The parameter that we use is the Cmax, Tmax, and area under the curve. As you see in this graph, it will be easy to understand about this is the original drugs and this is the generic drug that they try to build up to be equal as the original. But this one is not equal. We cannot approve this one to equal the original one. Okay. And when we perform the drugs product and we do the clinical trial in vivo and in vitro study already, we have to looking back to the product quality also, like how we keep the chemical to be stable, how we keep the drugs like a sterilization, and how to control the potency of the drugs, and how to validate the manufacturing process. The like a GMP will be considered in the pharmaceutical company. So it means that if you would like to start to produce or to develop the drugs, you have to keep your mind that what kind of the target animals or target species that you concern, and the target organ or the with disease or some protects that you like to do. Then you're looking for the formula with the fairly for using and fairly to keep in the quality control and stability. And you can looking for the method to testing and also concern about the toxicity effects. Finally, all that should be approved under your government or under the regulatory authority. And you can trust to use this. And we can consider the side effect that proved by the government or proved by FDA. Okay, so this is all my talks. Thank you so much. Hope
Thank
you
you.
can understand my language. Sorry for that.
Okay, thank you, Dr. Ason, for interesting presentations. Now we are going to the Q&A sessions. Due to the limitation of times, I think we just have one question for you, The question is, what are the key challenges in integrating pharmacokinetic and toxicokinetic data from preclinical models to human clinical trials, and how can these challenges be addressed?
You mean in the human trial, right?
Yep, yep,
Okay, this is almost depend on the approval, like ethic approval. Nowadays, when the when the company try to build up the new drugs or trial in the human, they have you have to concern about the ethic approval in animals together, like in animal. So you have to consider about the human consent for first before you file. Anyway, so I think should we start from the animal trial first or in vivo start in vitro study first, and then we can decrease the number of human trial. And this is catch your
yep,
your
yep.
question.
I think we just have one question for these sessions, and for another questions, we will collect the questions, and we're gonna send an email to you, and we will deliver back to the audience. I think
Okay,
this
thank
one due to the
you.
limitation of the times. Okay, I think that's all, Dr. Asons. Now we are going to the certificate awarding.
Okay, before ending the session, so we would like to give a certificate of appreciation to Dr. Akson. Yeah, doctor, please receive the certificate of appreciation from us. And once again, thank you for sharing your knowledge with us.
And at the end of these sessions, we would like to thank to all speakers for the informative and interesting presentations, and all the participants for the participations. And now we will give the sessions back to the MCs.
Good afternoon.
Wassalamualaikum
Wassalamualaikum warahmatullahi
wabarakatuh.
wabarakatuh.
Okay, for the next agenda, we will play a few poster presentation from our amazing student and lecturer. We hope you enjoy.
Now we are going to take a short break, but after that we will have our next session, which will be divided into 3 rooms. The first room will be moderated by Dr. Anissa Proboningrat, Doctor of Veterinary Medicine, Master of Science, and Dr. Hartanto Mulyo Raharjo, Doctor of Veterinary Medicine, Master of Science. Room 1 consists of topics regarding One Health, with the speakers Professor Dr. Lucia Trisuwanti, Doctor of Veterinary Medicine, Master of Agriculture from the Faculty of Veterinary Medicine, Universitas Airlangga; Ahmad Kurniawan, Doctor of Veterinary Medicine, Master of Science, Doctor of Philosophy from Groningen University; Dr. Emmanuel Ukbonai Buke from Ebony State University; and Dr. Randir Singh from Guru Angad Dev Veterinary and Animal Sciences University.
The second room will be moderated by Dr. Nani Hidayanti, Doctor of Veterinary Medicine, and Dr. Zulfi Nur Amrina Rosyada, Bachelor of Animal Science, Master of Science. Room 2 consists of topic regarding animal husbandry, with the speakers Dr. Iswahyudi, Doctor of Veterinary Medicine, Master of Agriculture, as the head of the Animal Health Division of Provincial Livestock Office of East Java, Indonesia; Dr. Mulyoto Pangestu from Monash University, Australia; Professor Enjong Kim from Kyung Pok University; and Dr. Muhammad Lukman Nordin from University Malaysia, Kelantan.
The third room will be moderated by Dr. Anissa Proboningrat, Doctor of Veterinary Medicine, Master of Science, Dr. Joko Legowo, Doctor of Veterinary Medicine, Master of Health, and Mirza Atika Madarina Hisham, Doctor of Veterinary Medicine, Master of Science. Room 3 consists of topics regarding clinical science and pharmacy. With the speakers, Professor Dr. Hani Plumeriastuti, Doctor of Veterinary Medicine, Master of Science, Veterinary Pathologist from the Faculty of Veterinary Medicine, Universitas Airlangga; Professor Dr. Jalila Abu from University Putra Malaysia; Professor Dr. Jong Ho Park from Kangon National University; Professor Albert Tai Ching Liao from National Taiwan University; Dr. Andres Gambini from the University of Queensland, and Dr. Carlo Gigana from the University of the Philippines at Los Baños.
Now, we are going to take a short break. Thank you and see you soon.
Welcome back, everyone. It's time for our next session, which will be divided into 3 rooms. The first room will be moderated by Dr. Anissa Proboningrat, Doctor of Veterinary Medicine, Master of Science, and Dr. Hartanto Mulyo Raharjo, Doctor of Veterinary Medicine, Master of Science. Room 1 consists of topics regarding One Health, with the speakers Prof. Dr. Lucia Trisuanti, Doctor of Veterinary Medicine, Master of Agriculture from the Faculty of Veterinary Medicine, Universitas Airlangga, Ahmad Kurniawan, Doctor of Veterinary Medicine, Master of Science, Doctor of Philosophy from Groningen University, Dr. Emmanuel Ukbo Naibuke from Ebonyi State University, and Dr. Radhir Singh from Guru Angad Dev Veterinary and Animal Sciences University.
The second room will be moderated by Dr. Nani Hidayanti, Doctor of Veterinary Medicine, and Dr. Zulfi Nur Amrina Roshada, Bachelor of Animal Science, Master of Science. Room 2 consists of topic regarding animal husbandry, with the speaker, Dr. Iswahyudi, Doctor of Veterinary Medicine, Master of Agriculture, as the head of the Animal Health Division of Provincial Livestock Office East Java, Indonesia. Dr. Mulyoto Pangestu from Monash University, Australia, Professor Eun Jong Kim from Kyung Pok University, and Dr. Muhammad Lukman Nordin from University Malaysia, Kelantan.
The third room will be moderated by Dr. Anissa Proboningrat, Doctor of Veterinary Medicine, Master of Science, Dr. Joko Lugowo, Doctor of Veterinary Medicine, Master of Health, and Mirza Atikah Madarani Hisham, Doctor of Veterinary Medicine, Master of Science. Room 3 consists of topics regarding clinical science and pharmacy, with the speakers Professor Dr. Hani Plumeriastuti, Doctor of Veterinary Medicine, Master of Science, Veterinary Pathologist from the Faculty of Veterinary Medicine, Universitas Airlangga, Professor Dr. Jalila Abu from University Putra Malaysia, Professor Dr. Jong Ho Park from Kangwon National University, Professor Albert Tai Ching Liao from National Taiwan University, Dr. Andres Gambini from the University of Queensland, and Dr. Carlo Gicana from the University of the Philippines at Los Baños.
You may join a room with your desired topic. The link have been provided through the Icvmhs website and also on the Zoom meetings chat box.
Also, to all the oral presentation participants, you may join your respective rooms. The moderator in room 1 will be Lina Susanti, Doctor of Veterinary Medicine, Master of Science, Doctor of Philosophy.
The moderator in room 2 will be Tantri Diawidi, Doctor of Veterinary Medicine, Master of Science.
The moderator in room 3 will be Arindita Niatazia Novianti, Doctor of Veterinary Medicine, Master of Science.
To all the participants, you may join your preferred room. Thank you and see you soon.
Good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. Welcome to room 1 of today's conference, focusing on the theme of One Health. I'm Anissa Proboningrat, your moderator for this session. It's a pleasure to be here with you all. Without further ado, let's dive into today's topics. Before we begin, we would like to take a moment to introduce the distinguished background of our speaker. And our first speaker today is Professor Lucia Trisuanti. Professor Lucia has demonstrated a lifelong commitment to academic excellence and research. She earned her undergraduate degree in veterinary medicine from Gadjah Mada University, shaping her foundational skills from 1981 to 1987. Continuing her academic journey, she pursued a master's degree in veterinary science at Gadjah Mada University from 1993 to 1996. She furthered her academic pursuit by obtaining a doctorate in medicine science from Universitas Erlangga, completing her studies from 2001 to 2005. Professor Lucia is currently affiliated with the divisions of Veterinary Parasitology, Faculty of Veterinary Medicine, Universitas Erlangga. Her research and professional journey have encompassed significant contributions to the field of parasitology. Her dedications has resulted in 69 international publications and a Scopus H index of 7. Professor Lucia is an active member of several professional organizations. The first one, she has been a member of APARVI since 2014, and she also has been a member of P4I since 1990. Today, Professor Lucia will deliver a seminar titled Toxoplasmosis in Livestock in Indonesia. This presentation promises to shed light on a critical issue in veterinary parasitology. Participants, if you have any questions during the session, please feel free to use the chat box for the Q&A session following the presentations. And please join me in extending a warm welcome to Professor Lucia Triswanti. The session is now yours, and you have 35 minutes for your presentation, followed by a 5 minutes Q&A session.
Good morning, ladies and gentlemen. It is a pleasure to be here with you today. First of all, I would like to say thank you to the committee for inviting me to this international conference and for giving me the opportunity to talk about toxoplasmosis in livestock in Indonesia. Next. I would like to begin this presentation by giving you a brief outline of my presentation. First, introduction, and the second, I will like to describe life cycle of Toxoplasma gondii. After that, I will review clinical sign of toxoplasmosis in animals, and then I will show the prevalence of toxoplasmosis in livestock in Indonesia, and finally, conclusion. Now, we begin with introduction. Toxoplasmosis is zoonotic disease that caused by Toxoplasma gondii. Toxoplasma gondii is an obligate intracellular protozoan parasite belongs to phylum Apicomplexa. The disease widely infected humans and warm-blooded animals, including livestock. Cats and another feline are definitive hosts, while the other animals are intermediate hosts. Toxoplasma gondii are capable of proliferating within a variety of nucleated host cells, including lymphocytes, macrophage, liver cells, intestinal cells, muscle cell, neuron, and another cell types of central nervous system. Prevalence of toxoplasmosis in the world depends on the location and host species. Next. Toxoplasmosis has a significant impact on health of human, domestic animals, including livestock, wildlife, and even ecosystem. Thus, it is included as a One Health disease. Molan et al. 2019 described that in almost all continents, infections occur in people with varying seroprevalence. In Asia, including in Indonesia, the total seroprevalence was recorded at 60.4%. According to the result, a review by Papas et al. 2009, the seroprevalence of human toxoplasmosis in Indonesia is high. In this picture, Papas et al. Classified the seroprevalence toxoplasmosis into 6 groups based on prevalence rate. In this picture, group described with color. White color represents the area with absent data toxoplasmosis. The green color was less than 10%. The blue one was 10 until 20%. And the prevalence rate 20 until 40% was represented with yellow color. Light red equal with 40 until 60%. The highest the prevalence rate of toxoplasmosis, more than 60%, was described with dark red. Now, see in Indonesia. All of area are red, light red and dark red. It means the seroprevalence of toxoplasmosis in Indonesia more than 40% and were distributed at all of Indonesia. This data is accordance with research by Redmanasari et al. 2017 and Tunda et al. 2017, who conduct research in Central Java and North Sulawesi, respectively, finding a seroprevalence of 62.5% and 58.5%. Then the question is, how the human can be infected? Humans become infected by ingesting tissues in undercooked livestock meat or milk, or consuming food or drink contaminated with oocysts, or by accidentally ingesting oocysts from environment. Tissues can occur in any cell in infected animal tissue, including livestock, especially in muscle and brain. Oocysts are set in catfishes and can remain viable in soil and water for months to years. Understanding prevalence toxoplasmosis has importance for both human and animal health. Because of it, I chose the topic presentation today, toxoplasmosis in livestock in Indonesia. But before we continue discussing this topic, I would like to review the life cycle of Toxoplasma gondii first. Life cycle Toxoplasma gondii is very complicated. As a member phylum of Apicomplexa, Toxoplasma gondii has 3 infectious states: tachyzoid, tissuecil that contain the bradyzoid, and sporulated oocysts that contain the sporozoites. Tachyzoid is rapidly dividing invasive state, and they have size range between 3 to 7 microns in length and 2 until 4 microns in weight. This state is found in condition acute illness. Bradyzoid is slowly dividing state, and bradyzoid clustered to form a cyst in host cell and called tissue cyst. The size of tissue cyst varies between 12 until 100 microns, depending on the tissue it occupies. Oocyst is result of sexual development in the cat intestine and is set along with cat feces. And so it is called an environmental state. As I mentioned before, cats and member phyllite were definitive hosts. Now, I will describe life cycle Toxoplasma gondii in cats. Cats and definitive hosts are infected by eating raw meat or milk that containing tissue cysts or bradyzoite, or ingesting sporulated oocysts. Tissue cysts and oocysts rupture, releasing bradyzoite and sporozoite. Bradyzoite and sporozoite and then infect intestinal cell and undergo caesareogeny in cat's intestine cell. Rupture caeson releases merozoite and merozoite developed by gametogony undergo macro and microgametes. Microgametes fertilize the microgametes and produce oocysts. Gametogony and oocyst formation also take place in epithelium of small intestine cats. Unsporulated oocysts are released into the intestinal lumen and pass into the environment with the feces cat. Sporogony occurs outside in the host and leads to the development of infectious oocysts which contain 2 sporocytes, and each sporocyst contain 4 sporozoites. And how the life cycle of toxoplasmosis in livestock? If the sporulating oocysts are ingested by an intermediate host, in this case livestock, then Toxoplasma gondii develop asexually. The asexual component consists 2 distinct states. They are tachyzoid and bradyzoid. In intestine livestock, the oocyst rupture, releasing sporozoites, and the sporozoite penetrate the intestinal epithelium to the lamina propria and develop into tachyzoids. Tachyzoid multiply rapidly through repeated endodiogeny and spread to various type nucleotide host cell during the acute infection. In chronic infection, tachyzoid convert to bradyzoid. Bradyzoid multiply slowly through endodiogeny and form tissue cysts. Tissue cysts are very great in size and occur in variety of organ and cell types, but predominantly found in central nervous system, brain, and muscle tissue. Tissue cysts are terminal life cycle state in livestock. Let's see the clinical sign toxoplasmosis in animals. It is usually asymptomatic, but can be severe and even fatal to many hosts in any condition. For example, in pregnant sheep, goat, and pig, toxoplasmosis causes abortion, neonatal mortality, and another congenital defects. Common clinical manifestation toxoplasmosis in cats and dogs are pneumonia, hepatitis, and encephalitis. Especially in cats, diarrhea with or without blood are often seen. So, let's continue to the prevalence toxoplasmosis in livestock in Indonesia. I reviewed this prevalence data from paper both published in Indonesian national and international journal published from 1976 until 2023. The type of livestock that are object of research are goat, pig, sheep, cattle, buffalo, chicken, and ducks. Prevalence levels data vary depending on the type of livestock, method, location, and year of the research. The following is the prevalence of each livestock. First, the prevalent toxoplasmosis in goat. The prevalent toxoplasmosis in goat has a high number, with range 15 until 93.9%. Based on the location, toxoplasmosis predate out at Sumatra, Java, Kalimantan, and Bali. Most methods were serological, CATT, LET, MAT, DAT, IHE. And one study used the PCR method, and there are 2 surveys using the bioassay method. Bioassay method proves the presence of parasites in goat meat. Because we found parasitological prevalence in meat goat, we must be alert that it will be dangerous for us who like to consume under goat meat like satay or steak. Let's go to the prevalent toxoplasmosis in sheep. The prevalent toxoplasmosis in sheep quite high, but little bit lower than in goat. It is range from 10 until 82.03%. All of the research methods are serological, CATT, DAT, skin test, and 2 MIT. Research location mostly in Java Island and only one in Sumatra, in Banda Aceh. Let's see the prevalent toxoplasmosis in pig. It is also varied with range 2.3 until 75.19%. Like previous animal, the method mainly serological. LET, ELISA, DAT, and only one location used bioassay. As we can see from the table, toxoplasmosis in pig was spread out evenly to the island in Indonesia, Sumatra, Java, Kalimantan, Sulawesi, Bali, and Papua. The prevalent toxoplasmosis in cattle also varies, ranging from 1.59 to 92.65%, and was spread across the island of Java, Sumatra, and Bali. Research methods are all serological, CATT, LET, ELISA, DAT, and 2 MIT. Seroprevalence of toxoplasmosis in buffalo, there are only 2 data. Both are located in Sumatra, and the research was carried out serological with prevalence rate 15 and 27.3%. The poultry that are being studied for toxoplasmosis are chicken and duck. There are only 2 prevalence data on duck, both located in Sumatra using serological study method. The prevalence rate is 6.1 and 20%. Meanwhile, for chicken, there are 5 data spread across Sumatra, Java, especially Surabaya and Bali. The method used are serological and bioassay. The prevalence rate varies 3.8 until 30%. This map of distribution toxoplasmosis in livestock in Indonesia. If we look at the map, the yellow color represent area where toxoplasmosis infection has occurred in livestock. All of the island of Java and Bali are painted yellow, while on another island only some of them. The white color represent the absence of prevalence data. This is opportunity for researchers to conduct research in this area because one route of transmission toxoplasmosis in human is ingestion of tissues in meat and another livestock product. I think it is important to always carry out toxoplasmosis surveillance in livestock and their product. Finally, we come to the conclusion. The prevalent rate toxoplasmosis in livestock in Indonesia varied widely, ranging from 1.59 to 93.9%. Serological survey were the most method of research. The distribution ranges from Banda Aceh to Papua, but the data is concentrated on Java and Bali Island. Surveillance need to be carried out through Indonesia. I think that's all. Thank you for your attention.
Thank you, Professor Lucia, for your insightful presentation on toxoplasmosis in livestock in Indonesia. We will now open the floor for questions. Participants, please feel free to type your questions in the chat box. And before we proceed to the Q&A, I want to extend our sincere gratitude to Prof. Lucia for sharing her expertise and research with us today. Your dedication to advancing veterinary parasitology is truly inspiring. Okay, right now we have some questions for Professor Lucia, and I will read the first questions for you. The first one is, could you elaborate on the clinical signs and symptoms of toxoplasmosis in infected livestock? And Professor Lucia. Professor Lucia, can you hear me?
Okay, thank you for the question. As I said in the presentation, toxoplasmosis is asymptomatic disease, and so it is the cases in livestock. However, in pregnant cattle, it is will result in abortion, neonatal mortality, stillbirth, and also congenital abnormalities. I think it's
Yes, okay. Thank you very much, Prof. Lucia. And then for the second questions, what are the economic impacts of toxoplasmosis on the livestock industry?
okay. Thank you for the question, Dr. Anisha. The economic loss of toxoplasmosis in livestock in industry, I think it is a reproduction disorder because it's if cattle infect with toxoplasmosis during pregnancy, especially infection in the first trimester, it is abnormal pregnancy and as I said before, it is can abortion and also fetal mortality and stillbirth and also congenital abnormality.
Yes, thank you, Professor Lucia. And for the third questions, are there effective strategies for the prevention and control of toxoplasmosis in livestock populations?
Thank you for the interesting question. Because toxoplasmosis is transmitted to the livestock through oocysts, which contaminated in environment, and oocysts are excreted with cat feces. So I think the most effective prevention is to free livestock from stray cat. And this strategy has practices in America in pig farm, and it is successful. I think it's can imitate in Indonesia. I think.
Yeah, thank you very much, Professor Lucia. Actually, there is another questions, but before the limited time that we have, we should end the question and answer sessions. And I would like to inform Professor Lucia that a virtual certificate of appreciation will be presented to you for your valuable contributions. Please receive it, Professor Lucia.
Thank you. Thank you, Dr. Anisha.
You're welcome, Prof. Lucia. All right, now I have the pleasure of handing over the next session to the next moderator, Dr. Hartanto.
Hello everyone, good afternoon. Assalamualaikum warahmatullahi wabarakatuh. Thank you, Dr. Anise, and welcome to room 1 of today's conference for One Health Term. And my name is Hartanto Mulyara Harjo, and I will become your moderator for today for this session. And for the second topic, we have Dr. Ahmad Kurniawan joining us today. Dr. Kurniawan, are you with us? I think I saw you. Hello. Good afternoon, doctor. What time now in Europe? We cannot hear your voice, doctor. Okay. Can you?
Okay. Can you hear me now?
Okay, I can hear you now.
Sure. Sorry, I tried to unmute myself, but it's not working.
Okay.
Good morning. It's 7 o'clock now in Groningen.
Oh, Okay, it's early in the morning. Okay.
Yeah.
So, Doctor, before we start, I think I will read your curriculum vitae. Okay.
Yeah.
Okay, so this is the curriculum vitae of Dr. Kurniawan. So he is a PhD in nuclear medicine and molecular imaging 2023 until now. He is MSc in biomedical science, University of Glasgow 2021 until 2022. And then he's also Doctor of Veterinary Medicine and also Bachelor of Veterinary Science from Universitas Airlangga. And his research interest is radiobiology, targeted radionuclide, and therapy for cancer, especially signal transduction and molecular biology also. For his affiliation, he's part of radiopharmaceutical research group and then research center for radioisotope and then radiopharmaceutical and biodosimetry technology, National Research and Innovation Agency from January 1st, 2018 until 2023. His publication, he have international publication, 20 journal. So without further ado, Dr. Ahmad Kurniawan, a lecturer titled Optimizing Biorisk Management Strategies, Safeguarding Health and Environment. And if you have any question, you can use the chat box or raise your hand later on Q&A session. For Dr. Ahmad Kurniawan, the session is yours.
Yeah, thank you. I'll try to share my screen. Can you see it?
Yes, we can see.
Okay, thank you. Okay. It's quite early in the morning so good morning everyone and good afternoon I think in Indonesia. So currently I base in the Groningen, the northern part of Netherlands. So first of all, I want to say thank you very much to the committee of International Conference on the Vet Medicines and Health Science in Indonesia for inviting me. It's quite, I don't know, this topic is, I want to say this is complicated, but in implementations, but in terms of understanding, I think a lot of expertise also come from Indonesia for the safe biorisk management. So for today, I will try to give a short presentation. I think it will cover in general about optimizing a biorisk management strategy, especially about safeguarding health and environment. So this is the outline of my presentation today. I will start with, I think the moderator already explained about my personal introductions, and then I will continue about talking a little bit about biorisk management and then risk assessment and hazard of biological materials, followed up by the health and safety. And I will try to talk about waste management because it's quite important. And yep, this is me. Mostly, I spend my time to study after I graduate from Airlangga University in Surabaya. I study in the Europe. So mostly my presentation is based on my experience, the time I spend in Europe, either in the UK, when I do my master's at the Cancer Research Center, the Wolfson Walker Cancer Research Center in Glasgow, or currently I base in the University Hospital. So it's quite weird since I'm a vet, but mostly I spend my time at the university hospitals. So, but it is what it is. So because my research is mostly about cancers. And then I have several training, I think quite related with today's presentation. I do several training about the biorisk management recently at the University of Edinburgh in the UK. And then some training about lab animal science in Norway, in Netherlands, and other trainings this year. If you want to reach me, you can contact me by emails. The one is for my university current affiliations contact, and another one is my agency in Indonesia. So first of all, to start my talk today, I'll try to do a flashback about recent cases that raise significant interest in the biosafety world, I would say. There is a case of one of a scientist from top university in the Barcelona. from Spain. From Universitat de Barcelona, there is one scientist with 45 years old, which is infected by the Creutzfeldt-Jakob disease. It's still under investigation, but it has raised significant awareness from the community since someone who should be careful, I mean, with a highly protected laboratory, but they could get infected with this kind of this is a prion. So there is something need to be evaluated on the how we work with the biological agents. And then when we try to going back in 2014, there is a reports from CDC in Atlanta, one of the biggest lab in Atlanta. There is a potential Ebola exposure to laboratory workers. And when we try to going back again in 2003, sorry, there is a SARS cases in the lab worker in Taiwan, in China, but this is also still under investigations. Based on all of this, I think what we try to give a concern is how we need to raise an awareness, even in the lab with a highly protected, with high facility, there's still a chance for us, for a scientist especially, to get a chance for infections or exposure from the biological agents. So when we try to talk about the biorisk management, I think it's better to start with definition of biorisk itself. Is some keywords is come up, start from the combinations of probability of occurrence of harm and severity of the harm, where the source of harm is a biological agents. So when we work with biological agent or toxins, and there is, we try to put into context, there is a probability of occurrence and the severity, and then we will define that as a biorisk. So there is a risk that we need to identify at the first, and then after we identify that risk, we need to evaluate the risk itself and try to determine the controls, the measurements. What should we do to control all the biorisks, and try to implement that in our facility. And then after we implement it, we need to know how successful that are by review the effectiveness of our controls, control implementation in our facility. There is several guidance on the biorisk management, which is, I think it is used worldwide, even in Indonesia too. There is a CWA 15793, the guidelines implementation of CWA, and especially the ISO 35001, especially for the biorisk management for laboratories, which is become the guidelines that we must have in our facility. And today I will try to talk a little bit generals about the risk assessment and hazard of biological materials. When we talked previously about the biorisk itself, it starts with biological agent and toxin. The biological agents, and then when the biological agent we use in our lab, we also use the cell culture, either to culture the cells or bacteria and etc. And also try to include the radioactive waste here, even though this is not really included into the part of biological materials. But in some experiments, you know that there is a combination of this. Like in our facility, we are using biological agents, we also using cell cultures, and we are using radioactive materials. So the risk assessment for these three different type of materials is also different. Either we are used as just a biological agents, but when we culture them and we combine with radioactive materials, so there is a three different assessment that we need to perform before we start our experiment, so we can make a good control measurement. And for biological agent itself, there is a lot of manual, but one of the most used in the field is Lab Biosafety Manual 4th editions. You can freely download it. I put on the link from my slides. That's from the WHO, which is actually cover all the aspect of biological agents. But in this presentation, since of the time limitation, mostly I will concern about the biosafety cabinets and other containment device, and some about the waste requirement. But overall, if you want to go deep about what is biological agent, how to perform the risk assessment, how to prepare the PPE, the proper PPE for our lab experiments, and how to prepare the outbreak preparedness and resilience, and etc. You can go just to the manuals, and it is cover all of the aspects. And to start about the biological agents, route of infections, if you are working on the lab, mostly there's 4 route of infection that we need to be aware. I think we all know all of this, but sometimes when we are working, I think regularly or more often in the lab, we start to be like not aware about all of these route of infections. First, airborne or inhalation into our lungs. That's why in the labs, we need to wear the mask or proper PPE, depends on the type of risk agent that we are working on. So why we need to wear a mask in the lab? Because we want to prevent the inhalation into our lungs, and then try to avoid we as a lab worker spread something to our precious, I want to say, precious lab materials. And then permeations or indirectly through the membrane. So there is a chance of our biological agents indirectly penetrate our membrane. Let's say in this picture, you can see goes through the eyes. That's why you need to wear the goggles in the lab. That's like a mandatory. If you're working in the lab, either ML1 or ML2 lab, you need to wear the biosafety glasses too. And then percutaneous, directly into the blood. That's why in the lab, you need to put the containment, spirit containment for the sharp needles or the sharp materials. You do not put that together with the common waste, because there is a chance when you touch something, you grab something from that waste, you touch some, you get directly penetrate into your skin and goes into the blood, and try to think if that is biological agents or infectious agents. So that will be a huge issue. And then another route of infection is ingestions into the gastrointestinal tract. That's why mostly in the lab, I think, especially in the university lab, students not really aware. They are sometimes eat in the labs, but this will be not happen, especially in the microbiological lab or in all type of lab, microbiology, also maybe in the fed science, in the fed public health and everything. You need to avoid eating on the lab. There is a huge chance of contamination. Maybe you can say that, oh, my hand is clean or something like that, but there is always a chance of contamination by, let's say you are not aware in the lab by eat or something else, or eat or drinks or something like that. This all of risk and this all of route of infection, I think, should be become awareness for the whole lab, and especially with the lab with a lot of visitors, and become the main rules that need to be applied. And then I'll try to talk about the when we already know what kind of route of infections, we try to classify the risk group, so we aware how to deal with. There is a 4 risk group that's that basically classified based on the human pathogens. The risk group 1 is all of the biological agents which is not cause a human disease risk. So in risk group 1, I put that on green. Yes, you can use that on the lab which not really contained with highly contained facility, but you still need to be aware even that is a risk group 1. And then move to risk group 2, the biological agent that can cause a human disease and may be hazard to the employee, but it is unlikely to spread to the community. And there is a usually effective treatment was available. It is almost like in the risk group 2, the biological agent also can cause severe human disease and might present a risk of spreading to community and there is a usually effective prophylaxis or treatment available. But this has different is it might present a risk of spreading to the community. And risk group 2 is unlikely to spread the community. So you need to remember that point. And risk group 4, this is the most risk group, which could suffer human disease in a serious hazard to employee, is likely to spread to the community, no effective prophylaxis. Let's say if you mentioned about Ebola, or previously before the treatment of COVID has invented, they also it's at least a movement between risk group 4 and risk group 3. But mostly if there is a new pathogen and there is no treatment and it will be spread to the community, it will be classified to risk group 4. And try to move a little bit, since I already talked about the biological agents. I'll try to talk about the cell cultures, since it's always related if you are in the lab. The cell culture itself, mostly there is a lot of factors to be considered for the risk assessment. If you are working with the cell cultures, first, you need to know the species. If you're working with the human cells, it is the risk is you get more risk if you compare with you are working at a primate, and if you're working with primate, you have more higher risk compared with another mammalians. Let's say like you are working with the mice cell lines or rat cell lines, and you have less risk factor to consider for the risk assessment. And then the tissue type. The blood has a higher risk compared with lymphoid and then neuronals and spleen, liver, kidney, because there is some more chance of the risk for us as a lab worker to get let's say a chance to get an exposure from the samples that we are working on. And then the source population is also a factor to consider, and then the type of cell cultures. If you take the cell lines from the hospitals, from the patients, and try to make your own primary cell cultures, let's say if you take from the breast cancer patient, ovarian cancer patients, and then you culture the cells, compare with you just buy from the vendors, let's say like ATCC or ICAC, the chance of you to get to consider into high risk is the primary cell cultures, because you never knows there is some virus in there, some infectious disease in there, or there is a lot of things that we are not really well characterized from our primary cell cultures. And the last but not least is the culture mediums. It's mostly if you're working on the cell cultures, you are using a fetal bovine serums, which is also a good medium for the bacteria or for virus to grow. And this is also taking into considerations. And I put on the right here, this is one of examples that what we are doing on the lab after we are working with the cell cultures, if we are put the disinfectants or virucides into our used mediums or used cells. So before we collect it, but taking into account, do not throw away into your sinks after you are working. Even you already put the vircons or disinfectant in there, you need to collect it and separate it. I will discuss later on the waste management. And this is the biosafety level. In some countries, like in Netherlands, we are more often use the term microbiological levels, ML1, ML2. But it's almost the same when we are talking about the biosafety level 1, biosafety level 2, and 3 and 4. It's always related with the risk group that which kind of pathogen that we are working on. And come to another of factors that we need to consider is disinfection and sterilizations. When we talk about disinfections, it is mostly about eliminates almost all microorganisms, but not the spores. So how to make the item safe to handle, and by use or dispose when we can use the methods like chemicals, heat, filtrations. But when we talk about sterilizations, we eliminate all the microorganisms and the sporicidals. And the microorganism we expect will not survive by using chemical, steam, or heat. So when you talk disinfections, it's quite different because sterilization is all clean, nothing in there that we make sure by the chemical or steam. And it is always need to perform, especially when you are working on the lab, either your bench or your cell culture hoods or anything that you're working on, you need to do a disinfections before to make sure that your working area is clean. This is simple, but sometimes people forget it if they are working too often. And there is an alternative decontamination methods, especially when you go to cell culture food, you have the UV light inside, and it just also goes to the GMP facility. They put the UV light with certain length to make sure that all the spot in the inside of the lab is already decontaminated from the microorganisms. And this is the biological safety cabinet that we already talked a lot before, which is mostly divided by 3, class 1, class 2, and class 3. And class 1 is you can't protect your product, but you're able to protect yourself as a worker, and you protect the environment by the HEPA filter in there. But in class 2, you protect the product, you protect you are as a lab worker, and you protect the environment because we have a double HEPA filter. And class 2 itself, it is divided by class A1, A2, B1, and B2, which is different based on the X house level. Some is like 100% X house, like in B2, and B1 is 70% 30, and A2 is also different with A1. And for class 3, it is a full containment. It is mostly used in the microbiological level 3 or BSL 3 up to 4 in the class 3, which is fully contained, and it's able to protect the product, the environment, and also the worker, because we are using glove boxes. Nothing came out from the inside of the hood after we already working inside. And this is also one of part of the thing that I need to explain is how we make sure our hood is our working area is correct. So we always say this is a clean to dirty. But it is also depends on how you are, either you are right-handed or left-handed. For right-handed persons, is usually left to the right. So you put all the clean stuff on the left, and your working area is on the middles, and all your dirty stuff on the right. So you not mix it. So otherwise you get the cross contaminations, and that's also not affect your work, but also will affect you as a lab worker, and also the environment, because either you create more waste because you get more contaminated materials, or otherwise you get spreading contamination to another workers. I think that's mostly about the three different things. I will not discuss about the third part, which is the radioactive waste, since that will be taken more times. But you need to taking into account, as long as we put the correct control measurements and correct analysis of which risk group we are working on and how we work, is it correct or not, put a correct disinfection and sterilization, etc. You are trying to minimize the exposure either to lab worker or to the environments if you're working in the correct pattern. And then about the health and safety, mostly about the accidents and incidents, biosecurity, lab management, and occupational health. First, about the accident and incidents. When we talk about the accident, it's something that unintended event, which is giving rise to harm. And it is also very important that we take into account that incident or near miss, because simply accidents do not result in any actual damage. That's why I put two models in there. This is Swiss cheese models and domino effect. The Swiss cheese models is, you can see from this image, there is a different layer of cheese, but there is a hole in there. Different layer mean the control measurement that we already taking into account for our biorisk management, but we still have a hole, which means there is still a chance from the hazard to go through our control measurement. That's why we need to do the plan, how we do the do, act, and check, the PDAC for each step of our biorisk management. And then the domino effect is like one accident, unintended harm can lead to another cases, another huge, let's say if you are not sterilize your bench, it can lead you get contaminations. And let's say if you work in cell culture lab, your cell get contaminated, let's say from mycoplasma, and your cells contaminate your fellow cells inside of the same hood, and it will spread the whole lab, and it will ruin your experiments. That's why this kind of domino accident or incident is not really like just physical accident or incidents, but the whole thing. If you get contamination, if you get some accident or unintended event, you need to report it and set emergency planning. So your biosafety officer, if there is excess in your lab, they will take into account what should we do, what should we prevent, what should we do next. That's why I say this quite important. And then I will try, but I still have a time. I will try to talk about the biosafety and biosecurity a little bit. I think most of us already know. When we talk about biosafety, it's like more to containment measure taken to ensure the infectious agents can be safely manipulated by reduce the potential exposure to lab worker, us, people outside the lab, and the environment. So how we reduce the potential exposure to the lab worker, to the people outside the lab, and to the environment. This is different with biosecurity because when you talk about biosecurity, it's a steps which we take to ensure that certain infectious agents or biological agents or toxins not obtained by unlawful means. So we try to protect the agents against attempts that or theft or financial gains or potential threat posed by the terrorism. Let's in this picture, if you are working inside the lab hood and you are working with proper PPE and you already put all the step of measurement, you already use the biosafety correctly. It means you protect yourself, you protect the people outside by contain your biological agent inside of your biological containment, and you protect the environments because of your systems. But when you talk about biosecurity, it is start with personal security, how we go to the lab. You need to use your special card so you can enter the lab with a PIN code or something like that. You have a security in there 24 hours a day, in the 7 days to monitor everything that what's going on in the lab. You have a physical security, which is protect your building. And when you try to transport your biological material from one place to another, you have a transport security. That's how we work with the biological agent. You can't just let everyone goes in, goes out from your lab without thinking about anything because they don't know how it works. That's why we need to implement all of this. And then about the occupational health, it is mostly talk about the effect of work and health work. It is something that sometimes people forgot if we have the facility, which is working with biological materials. We need to concern about the vaccines for certain lab worker. If you have a lab animals, let's say, you need to take it into account, oh, my lab worker need to get these vaccinations. And because there is a chance like these animals, maybe we don't know the health status, we take it from different breeder and they can get a spread infection to my lab workers. So how you prevent that? That's why we talk about in the beginning. You need to give a vaccination to your lab workers. And then you need to do health assessment at least twice a year. You need to know your lab worker is in good health conditions. And you also need the ergonomics facility inside your lab. Let's say this micro pipette is better than using pipetting using a mouth, or your lab chair is not make your back is hard if you are using the good one. Either you're just using this wooden chairs, which is not good for your lab worker, I mean. That's why in your biorisk management, you are not only the commitment from your head of your department or head of your agency, but you need to also taking into account, you put the biosafety officer in there, you have the occupational health, which is also work together to implement all of the aspects to make this work-life balance for your lab worker and for your facility. So all of the aspect is in balance. I think now you should start to check in your lab how good you implement all of this. I mean, if you can try to improve it, not like drastically, but little by little, you can improve it. So it will affect your result at the end, because if you implement all these aspects. And I think this is one of the important thing I try to talk at the end about the waste management. Since I don't know in Indonesia how we regulate it, because especially till now, I think we are not really aware about separate the waste based on the category. Because usually there is always a third party who work to collect all of the waste, as far as I know. But in Europe, mostly, either in the UK or Netherlands, we have a solid ML1, which is, you can see the yellow bin here. There is always a sticker, let's say UN3245, which is marked as a GMO waste for road transport. And in my lab, mostly we are using the code 32191, which is solid ML2. This is used for pathogenic materials because we are working in the hospitals. So how we work, we separate the waste based on the category and we put the correct stickers. So the third party is just like, they will not accept if let's say we are not put this in the correct stickers, we are not put the waste in the correct bin, and we are not put this in the safety manners, let's say like the lid is still open, something like that, they will refuse it. That's how the management work. You need to make a SOP how we manage this waste. And this is an example of waste management applications that we have in our facility. This is for the liquid, let's say in the black mark in the stickers. This is for watery solution with pH more than 5, less than 5, blank marking, red marking means organic solvents, blue marking means organic halogen rich, and green make for oil and grease from workshop, something like that. So we really divided all the waste based on the pH, is this organic solvents or not, this is a halogens, because it is not only make easier for the waste collections and the waste management and the waste recycling after this, but it is also important about the containment itself, because you cannot put the organic solvent and organic halogen in the same place, otherwise it will be explode, something like that. That's why I think start from now, this is one of the most I want to encourage, either in Indonesia or in other country. We try to implement especially all of these aspects. Biorisk management is very complex. You are not only talk about this in 1 week, but especially the training itself consists of like 3 months. But in 1 day, I want to say that all of these aspects need to be implemented in the whole, and it needs like the will of our department, of our agency to implement all the aspects. And there is a Biorisk Management Biosafety Association now worldwide. In Europe, we have a EBSA.
Yes.
And in Asian Pacific, I think you have APBA.
Doctor,
and
can you hear me? Okay.
Yeah, this is my last slide.
Oh, okay, okay.
Yeah,
you can
this is
continue.
I think this is the last is International Federation of Biosafety Association. This is non-governmental associations. which is also offer professional certifications. And I also encourage a lot of staff from university to follow the biorisk training course. One of them at the University of Edinburgh. It's quite cheap because if you come from developed country, you get a discount, something like that. And you can follow the professional course in biorisk management. And they offer like online training too. I try to endorse that one. And this is a take-home message from me. Biorisk management requires commitment from the entire organizations. And plan, do, check, action is a key point for biorisk management implementations. And all staff and employees who work with biohazard agents are required to undergo training and supervisions. Otherwise, they don't know what to do. Before your staff go to work in ML1 lab, let's say, they need to know what is the SOP, how they work, what kind of biorisk they are dealing with, and etc. and etc. When you go to ML2 or ML3, the risk is increased, so you need more training for your staff. And this is the key reference. You can download it freely from the internet because the guideline is available. And I think that's all. Thank you very much.
Okay, thank you so much, Dr. Kurniawan. What a great presentation. And I agree with you that biorisk management is very important, and we know that a lot of people still not aware about this one. So it's actually very important things to do and important things to know, something like that. So Dr. Kurniawan, in here we already have some question for you, and the first question is from our student. He asked about what are the challenges and best practices in balancing the need for scientific research and innovation with the imperative to manage and minimize biorisk?
Yeah, thank you. That's a good question. Yeah. So basically, I know we have, I try to be honest. In Indonesia, we have an issue with the budget. We have a very small budget for our research, to be honest. We don't have like in Europe. You spend like €1 billion and you get nothing, just one paper, it's fine. In Indonesia, you get 100 million rupiahs and they ask about a lot of things. That's a fact. Try to be honest. But if you ask me how to balance this, I mean, you can't put the biorisk like something the third number or fifth number of your priority. You need to put the biorisk management, the biorisk approach, the biorisk control and everything at the first place first before you start your experiments. I mean, if you want to work with, let's say, zoonotic disease agent, which is already explained by by Professor Lucia before, like mycoplasma, you need to know which risk group it's the toxoplasma, which risk group it belongs. And then you need to prepare proper PPE and you need to have a good facility. If you don't have enough budget for that, so don't do the research, in my opinion. Otherwise, you just risk yourself, risk the another people in the lab, risk the environment by spreading a lot of things which is uncontrollable. That's why I always say that put the biorisk first. You can't be balanced if you have less budget. If you have a lot of budgets, you need to do the risk assessment first. You need to measure what kind of risk. You need to put the control plan, so you can put all of your money to buy the personal protective equipment. You try to do all the plan before you do the research. So that will be my answer. You can't just make it balance 100% if you don't really have enough budget for that. But if you have enough budget, put the biorisk on the first place before you start all of your experiment.
Actually, I very agree with you, Doctor. It's very important. Put biorisk first before we start our research. It's very important. So, Doctor, we still have, maybe this one is the last question before we close the session with you. So the next question is from Diandra. She asks about how can interdisciplinary collaboration enhance the effectiveness of biorisk management strategies in mitigating the impact of biological hazard?
Yeah, I try to get your point. I mean, all the research mostly involve interdisciplinary science. I try to get example from me because I come from vet background, and then I do my research in hospital. We have a chemist, we have a medical pieces, we have a medic, we have a specialist, we have a biologist, we have molecular biologist. I mean, don't take that as an issue. That is a good part because different people can give you different point of view. Maybe me, let's say I'm a vet, I don't really know about the risks that potentially come from, let's say, from organic chemistry. So that's why when we try to arrange the first place to analyze our biorisk assessment for our facility, for our research, for our project, all the people will sit together. They will give the input based on the point of view, based on the expertise. So that's why interdisciplinary science actually make the implementation of biorisk assessment itself is better than just one people like creating the document, because one people never know everything. We need another people to give a different point of view for us. So we can take it into account, oh, in the chemistry aspect, we cannot use this kind of liquid, something like that from the molecular biology, or this is have an effect for this, for our gene or DNA, something like that. And for our point of view as a vet, we have this point of view and etc. and etc. When try to elaborate all of that, I think this will be a beneficial for to create a proper biorisk assessment document, which will be implemented for our further research or project.
Okay.
I think that will be my comment.
Okay, and doctor, I think this is we still have questions. So this is
Yeah,
from
sure.
from Zoom. It's from Sally. My question for Dr. Ahmad: how to prevent the spread of dangerous agents in the laboratory when the workspace is close to the test room, where sometimes there are cooking and eating activities in the workroom. Oh, I think this one is common
Yeah.
in Indonesia, maybe. Yeah. So does the workspace have to be a separate building, something like that? So what do you think?
Yeah, I will try to give an example from my facility in the Cancer Research Center in Glasgow in the UK. So basically, we put a very big sticker in front of our lab. There is no food and drink goes inside the lab. So the workspace is only different room, but to open our laptop, just to work, no eat and drink. If you want to eat and drink, you have a pantry outside. Otherwise, you will just increase the chance because you never know. I mean, maybe you can put some, how to say that, some spot to clean your hands with disinfection before you enter the workspace. But you never know. Some people, maybe sometimes if they are regularly do that, they start to forget. And my advice is just do not eat and drink inside the lab, even if the workspace is stick in one building. Make it different locations. That's we already implement in the Europe. In the past, there is some labs, even in the Europe, they do like that. But now it's all different space because we try to avoid the risk, like the forward of infection that I already mentioned.
Doctor,
And can I answer the last question quickly?
Oh yeah. You can. You can. The last question, you can
Yeah,
answer directly.
about the economic benefit of waste management. I mean, waste management is very expensive. Do you know how much we cost for one bin? The blue bin is 50 euro. It's almost 1 million rupiah for just one bin. In one day, we produce like 10 to 12.
Yeah.
That's a lot of money,
Yeah.
to be honest, even in Europe. But what is the economic benefit? I mean, in science, we try to protect the environment. That's why we work. And we try to reduce the risk. And without good waste management, you just create another issue. First, we have already an issue with the microplastics, which is now we try to solve. But if you create another issue with not manage your waste properly, you already create another issue for the environment. I know this is a hypocrite because if you don't have any budget, so you say that, okay, we can just throw it away to the sinks our cell cultures medium. But try to think, you need to put your budget in the first place for all of this. You need to calculate how much you will spend for the waste management. Otherwise, we will not improve. We will try all our best to work on this, but it needs all the collaboration from the whole, whole stakeholders around the world to improve all of this. I think that will be my comment.
That's very nice, Dr. Kurniawan.
Yeah.
We learned something new today. It's very good. And
Yeah.
thank you so much for
Thank you
your
for the
times.
opportunity.
Yeah, and before we close the session with you, we have a token of appreciation from our faculty. So the certificate for Dr. Kurniawan. So Doctor,
Thank
this
you.
is for you. Thank you so much
Thank
for
you.
your
Thank you
time.
very much.
Thank you. And I hope
See you.
we can see you again in
Yeah, sure.
next
Say
event.
hello when you are guys visit Groningen or you go to Europe. You can
Okay,
just pass
for
through
sure, we
to our
will.
facility.
Thank you so much, Doctor. See you. Thank you.
Yeah. See you.
Okay, so
Have a nice day.
next we will go to the next presenter. The next topic we have is from Dr. Emmanuel Ugbo Nanabuike. He already joined with us today. Doctor, how are you? Can you hear me? Are you here, Doctor? Dr. Ugbo?
Hello, good morning.
Okay, Doctor, we can hear your
Hello.
voice. What time right now, Doctor?
Can you hear me?
Yes, yes, we can hear
Hello.
you.
Yeah, my network is very poor.
Ah,
Can you hear me now?
actually, the network is not
Hello?
good. Hello, Doctor. Yes, we can hear you. Maybe you can close your video. Yeah, some like this.
Okay, can you hear me now?
Yes, yes, yes. We can hear you clearly right now. Okay, so
Yeah, I've closed the video.
yeah. We can. And Doctor, before we start, I think
Okay.
I will read your curriculum vitae first. So the curriculum of Dr. Nanabuike Ugbo. So Dr. Ugbo is a Bachelor of Science, BSc in Applied Microbiology from Ebonyi State University, and then Master of Science in Medical Microbiology in Ebonyi State University, and also Doctor of Philosophy in Medical Microbiology from Nanamdi Azilke University. And his research interest is antimicrobial resistance in human and animals. Affiliation, he is a senior lecturer from Ebonyi State University. And for publication, he have a lot of publication, 68 journals, and the Scopus index is 11. Okay, so Dr. Ugbo, without further ado, today he will have a lecture about the combating antimicrobial resistance, strategies, challenge, and future director. So, Doctor, the time is yours.
Good afternoon, my distinguished colleague and the organizers of this conference. Thank you for having me. Today, I'll be making a presentation on combating antimicrobial resistance, strategies, challenges, future directions. First of all, we should look at what we understand by antimicrobial. Antimicrobial is antiviral, antifungal, parasites that are medicine used to prevent and treat infections in human and animal and plants. The effectiveness is now in jeopardy because number of these antimicrobial treatments that once worked no longer work again because microorganism has become resistant to them. And resistance
So, Dr. Ugbo, are you still with us? I think there's a connection problem with Dr. Ugbo. So he disconnect right now. So we will waiting for him to come back.
Hello, doctor.
Hello, yes, doctor. We can hear you right now. Okay, so you can start.
Can you hear me?
Yes, yes, we can hear you.
Sorry, it's raining heavily here, so it's
Yeah.
affecting network.
Okay.
Can you hear me?
Okay, doctor. Yes, we can hear you. Can you make
Can
it?
you hear me now?
Yes, we can hear you, doctor, clearly. And can you make it slideshow, doctor?
Okay.
Slideshow.
You say?
Yes, yes, we can see your PowerPoint. Okay, you can continue, doctor.
Can you see my slide?
Yes, yes, we can hear you clearly. Okay.
Okay, can you see it right?
Yes.
It's okay. Sorry, it's raining heavily here, so it's okay. We understand by what God to my bacteria, but even death. Who so not live resistance. Not resistance one and have one. That is when there is not to end in
Doctor, we cannot hear you clearly in here.
there is no any all
Doctor, can you hear me? Because we cannot hear you clearly. Is it okay if we are the one that share your PowerPoint from here, so you don't have to share your PowerPoint? Is it okay?
for kind. I can't do more than what I can do.
So looks like Dr. Ugbo have a problem with the connection.
No, they want to share the network. It's not going. Okay, actually share the network. Share. No, they want to share the PowerPoint. The man present is now. Can you on the Zoom?
Dr. Ugbo, are you still with us? Can you hear our voice? Okay, so I think there have some problem with Dr. Ugbo. Maybe we can start first with Dr. Randir Singh. Dr. Randir, are you with us? Dr. Randir, can you hear my voice?
Hello? Hello? Hello?
Hello, Dr. Ugbo.
Hello?
Yeah, I was unmuted. So
yeah, doctor. Thank you.
Can you hear me now?
Can you hear me now?
Okay,
Yes,
we can
I
hear
can.
you.
Hello?
So,
Yes, I can.
Dr. Ugbo, are you with us
Sorry
already?
for.
Okay, so Dr. Ugbo, is it okay if we start your, we will share the PowerPoint from here? Are
I
you
will
okay?
start. You
Are you
can
okay?
share the PowerPoint. Share it from there.
Yes, we will share the PowerPoint from here, so you can just tell us when we move the slide. Is it okay?
It's okay. It's
Okay.
okay.
Okay, doctor. So, can the committee help me to share the PowerPoint of Dr. Ugbo? Okay, doctor. So you can start.
Okay. Next page.
Next page.
Yes. It's very good. Okay. Like I said, I'll be talking on combating antimicrobial resistance, strategies, challenges, and future direction. Antimicrobial resistance. Antimicrobial includes antibiotics or antibacterial, antiviral, antifungi, and antiparasites. They are medicine used in preventing and treating human and animal and plants. And today, the effectiveness is in jeopardy as a result of, and also as a result of abuse. And antimicrobial resistance affects different countries in a varying ways. Now, before we progress, we need to understand what is called antimicrobial resistance. Next slide. Next slide. Okay. So antimicrobial resistance occur when antimicrobial resistance occur when bacteria, virus, fungi, parasite change over time and no longer respond to medicine. And antimicrobial and we have different types of antimicrobial resistance. One, multi-drug resistance. This occurs when organism is non-sensitive to at least one or more agents in 3 or more antimicrobial categories. And we also have another one that is called the extreme drug resistance. Non-resistance to at least one agent in all categories except in 2 or less. We also have sensitive to at least one agent in all antimicrobial categories. Next slide. Next slide, please. Yeah, we have factors that contribute to antimicrobial resistance. One of the factors is environmental factor, drug-related factor, patient-related factor, and physicians-related factors. Under environmental factor, overcrowdedness, poor sanitation, ineffective infection control program, or increased national and international travel. This occurs when people travel from one country to the other, thereby bringing in one or two resistant organisms to the other animal husbandry and agriculture as a growth promoter. You know, today, a lot of antimicrobial agents are being used. All these things are some of the environmental factors that is contributing to antimicrobial resistance. Next slide. Next slide. Yes, the next one is drug-related factor. So the drug-related factor is a situation whereby fake drugs are being distributed in the market and quality of the drug are low, especially when people go to the companies that produce drug, tell them to produce a low quality drug, then help them to make more gain. All these things encourages drug resistance in the environment. And patient-related factor, poverty, poor concept or lack of sanitation, self-medication. Today, you can bear me witness that many people intend to treat themselves when they are sick instead of going to hospital. All these things encourages drug resistance. And lack of education is one of it. Poor advancement of dosage regimen. Sometimes when we take antibiotics, we are sick. Instead of us completing our regimen, because we felt that we are getting better, we drop our regimen. All these things is also affecting antimicrobial, also contributing to antimicrobial resistance. So next one is physician's prescription. Next slide. Next slide. Yes, physician's prescription. Inadequate dosage, improper usage of available drugs, that is blind treatment, and overusage of antimicrobial, and lack of training, and current knowledge. Sometimes doctors don't actually go to get a current training and current treatment of antimicrobial agent. There are so many other modes of type of animals that are sick. Sometimes we treat them with antibiotics. We doesn't allow the animal to get healed completely. can be slaughtered and consumed use of the antimicrobial agents that is still remaining in the animal into human. By so doing, you also encourage antimicrobial resistance. Next slide. Next slide. Antimicrobial resistance. Antimicrobial resistance can actually occur by limit uptake of drug. It can also be in form of modification of drug targets or inactivation of a drug, and also active mechanism whereby organism uses to cause antimicrobial resistance. And today, the global impact of antimicrobial resistance has been estimated to have affect over 1.2 million people annually die of antimicrobial related disease. And it has been predicted that if it is unchecked by 2050, over 10 million people in the whole world on diseases related to antimicrobial resistance. Next slide. Next slide. Thank you. So there are some global impacts of antimicrobial resistance, such as one, in every death, one person died every 3 minutes in the whole world in a disease related to antimicrobial resistance. And today, there are some drivers of antimicrobial resistance to One Health. One of the drivers is misuse of antimicrobials in human. And another one is misuse of or overuse of antimicrobial in aquaculture. Because today, so many people are using antimicrobial in their aquaculture system, even when it is not approved. And the next one is poor infection and disease prevention and control in food and feeds of animals, and misuse or overuse of antimicrobial in our plant or crops. Today, we intend to use so many antimicrobial agents in the growth of our crop to enhance yields, or even to increase the rapture time. All those things also affect one of the drivers according to One Health. And another one is poor sanitation. And discharge of all these waste contribute to the dissemination of antimicrobial resistance in our environment. Now, next slide. How do you manage or respond to antimicrobial resistance? Environment play a key role in the development of antimicrobial and spread of antimicrobial resistance. Therefore, the response must be based on One Health approach, recognizing the human, the animal, plants, connected and indivisible. So at the global or regional or local level, from all the sectors, stakeholders, they have to come together to work in hand to fight this particular menace. So prevention is the core of the action needed in hitting the emergency of antimicrobial resistance, and environment is the key play in this particular situation. So environment is the key part of the solution. So we really need to care for our environment if we must combat antimicrobial resistance. Next slide. Yes, these are some of the approaches for combating antimicrobial resistance. So if you look at this organogram that I have here, it's just trying to tell you some things that we need to do to combat antimicrobial resistance in our environment. One, we have to increase collaboration We also make sure there is rational use of antimicrobial agents. Infectious control must be strong continuously, and there should be a ban in over-the-counter use of antimicrobial agents. Education and motivation must be incorporated into the people. Research, development of drug and the vaccine is very, very important. Immunization to cover areas that have been affected and that is prone to be affected is also very, very important. We also need to have what is called routine hand washing. So whenever you go out and come, you need to carrier of antimicrobial resistant organisms. So we also have to form a committees that will be monitoring the antimicrobial resistance. And we also have to have a national policy. Drugs that has to be used in a particular thing, both in animal husbandry, in the hospital for treatment of infection, and first week. Next slide. So there are some strategies which we can use to combat antimicrobial resistance. So one of the strategic area, number one, is awareness and education. So there must be a strong knowledge and evidence. There must be an improved awareness and understanding of antimicrobial through training and education. So microbiologists, epidemiologists, and public health officers should be able to know the basis of antimicrobial activities, basis of antimicrobial resistance, and source of antimicrobial immunism, transmission routes, and magnitude of the problem in the society, and be where to use, best way to use. Next slide. Strategy 2. The second strategy is strengthening knowledge and evidence through surveillance and research. Microbiologists and epidemiologists and public health officers are responsible for qualities of diagnosis, surveillance. They should be able to produce data, collection, and sort of resistant mechanism. They should be also able to report the source of antimicrobial resistance mechanism and detect, confirm, characterize communication of antimicrobial emergency if there is any. And they also be able to measure the impact through research and provide
Looks like Dr. Ugbo have a problem with the connection again. Let's wait for him for a moment. So with this, I also would like to remind you, we have room 2 and also room 3, which you can join. For the room 2, you can join for the animal husbandry term. And for the room 3, you can join for our clinical science and pharmacology term. So if you would like to join for another room, you can just join the Zoom link that we already provide to you. And while waiting for Dr. Ubo for the first room, we also have Dr. Randir Singh after Dr. Ubo. So please wait for a moment. Looks like we still cannot contact Dr. Ugbo because I already contact him, but uh, still cannot reach him. So, Dr. Randir Singh, are you with us right now? Dr. Randir?
Yes, yes, I'm there.
Oh yeah, okay, Dr. Randir, it's very good. It's very good to see you. Okay, so I think we will go for the next presenter while waiting for Dr. Ugbo. So we will continue with Dr. Ugbo later. So we will have present uh, from Dr. Randir first. So yeah, Dr. Randir, how are you?
I'm fine.
Oh, that's
Good.
very
Can you
good.
hear me? Is there any disturbance?
Yeah. We can hear you clearly in here. Dr. Randir, what time right now in India?
It is 12:30.
Okay, so
Yeah.
before we start, I think I will introduce Dr. Andir to you. So the curriculum vitae for Dr. Andir. Okay, so Dr. Andir Singh from Gatvasu. He's a BVSC from HNA College of Veterinary Science, India, and then MVSC from College of Veterinary Science, India, and he's also a PhD of Department of Biological Science from Clemson University, Clemson from USA, and his research interest is food safety, foodborne pathogens, and then antimicrobial resistant in foodborne pathogens, and then zoonotic disease, and bacterial molecular epidemiology. For his affiliation, Dr. Andir is a senior lecturer, and then for the publication
It is
have
wrong.
international
It is wrong.
publication 71 journal and book published is 7 and have its index is 16. So Dr. Andir today will deliver a lecture titled Zoonotic Disease in Asia, Challenge and Strategies for Border Control and Prevention. And during the lecture, if you have any question, you can use the chat box feature or ask the speaker later on Q&A session. So Dr. Andir, the time is yours.
Thank you. And I'm going to share my slide, but then there is one correction. My affiliation shown, my VT was not right. My affiliation is Center for one that they Veterinary and Animal Sciences University in India.
Doctor, we cannot hear your voice clearly.
Affiliation shown in my CV was not right.
Yeah, we are very
So
sorry, Doctor.
it is College of Veterinary Science, Bangladesh Veterinary Animal Sciences University.
Okay, thank you so much,
So
Doctor. We will correct it.
Copy paste mistake.
Okay, thank you.
Okay.
We are very sorry for the inconvenience.
So, now let me share my slide, and there it is. All right. First of all, I would like to extend my thanks and to the dean, Dr. Mirni Lamin, Faculty of Veterinary Medicine, University of Erlangen, and Dr. Hertanto for giving me this opportunity to this international conference. And today I'm going to share my views on zoonotic diseases in Asia, challenges and strategies for border control and prevention. And now, if you look at this picture, it reminds us of COVID-19 outbreak, which brought an entire world on its knees and led to extensive lockdown, a lot of morbidity and then mortality. And this reminds me of a famous quote from famous scientist Louis Pasteur, that it is the microbes who have the last word. And it is rightly said, because these microorganisms, they are the one who came first. They are the strong, they can easily evolve, they are well adapted, they have all the arsenals to fight us. So I think it holds good. Then it's in lighter note. So whosoever said one person can't change the world, never eat an understood that. Now this can relate well with the COVID-19 outbreak. We know that one virus has changed the entire world. Now, if we talk about zoonotic diseases, we know that this word zoonosis term was coined by Dr. Rudolf Virchow, a famous pathologist. And when he coined this term, this was the beginning of linkages between human and veterinary medicine. Now, zoonotic diseases, we know these are transmitted from animals to humans, poses significant public health threat globally. And Asia, being a hotspot for such diseases due to its diverse ecosystem, dense population, close interaction with humans and animals. Now, zoonotic transmission involves interaction of pathogens and at least two host species. First is the natural reservoir host infected with pathogen, often asymptomatic, shedding the pathogen. Second, a recipient host presenting the disease after infection with pathogen from different hosts. There may be involvement of third host as an intermediate host that may or may not be present, but act as bridging or mixing vessel. Can be vertebrate or invertebrate. Now, among the zoonotic infection, the one caused by viruses are the one that poses major threat to human health, as they can be transmitted by aerosol, direct contact with animal, or food, or their fluids, through food or through vector. It is estimated that more than 1.6 million unknown viral species of mammalian and waterfowl can infect humans, of which half of them have potential zoonotic infection. Now, in this scenario, you will see that the serious one is the one where zoonotic infection adapt to humans, and then it leads to human-to-human transmission. When it leads to human-to-human transmission, then it can lead to disaster, and this is what happened with COVID-19. human-to-human transmission. There are other diseases present out here which have potential for human-to-human transmission, and they are doing so. For example, Nipah virus, for example, Ebola virus. Now, as far as emerging diseases are concerned and their definition is concerned, they can be classified or defined in three different ways. One, those diseases that have not occurred before. Best example is COVID-19 outbreak, and those diseases which have occurred previously but affected only a small number of people or have occurred at isolated or small geographical area, but now their incidence or area of occurrence has increased. Or we can also say that those diseases which have occurred throughout the human history, but they have recently been recognized as an important infectious disease. We know that out of most 60% of the existing human infectious diseases, they are zoonotic in nature, and 75% of the emerging infectious diseases, they have origin in animal. It is said that every year 5 new human diseases appear, and out of those 3 they have origin in animal. And out of the available agent or pathogens with bioterrorist potentials, 80% of them, they are zoonotic pathogens. Now, why zoonotic diseases have emerged? Now, there are several factors. One is human-wildlife interaction. These zoonotic diseases, they are natural reservoir in this wildlife environment, where these diseases are maintained. But as a man, human has encroached upon this wildlife environment for the purpose of hunting, for the purpose of illegal trade of animals, for the purpose of clearing forests for agricultural land, for the purpose of recreational activity. They have broken this natural cycle that is occurring in the wild and has contracted the disease. Our disease has spilled to a human population. When the human came into existence, initially we were hunters. We gathered food. And as we progressed, we learned the art of domestication. We realized domestication, how this would help us. And then later on, it was supplemented by agriculture when humans started settling around water bodies. And then as we progressed, we had the benefit of industrialization, urbanization. All these activities further made or brought us in close contact with the wildlife population, because our need, our demand increased. And in these wildlife population or these areas, they are the one who had opportunities for us to read those demands. But that at the same time, it helped us to reach those natural phenomena that were occurring there and led to the spillover of disease. Then wildlife trade, hunting of animals indiscriminately for illegal trades or legal trades, has again brought us close contact with the animal. Then when human, as I said, human came into existence, and as population grew, the need of feeding that growing population arose, and we had limited land. In order to meet the grain or food demand of this growing population, we resorted to clearing forest. And when we started growing grains, we needed water for irrigations, and we started storing waters, canals, rivers, dams, and then these became source of breeding ground for mosquitoes and vector-borne diseases. As per some estimates, agriculture is the biggest user of water, and this can reach up to 90% of total water production. Then as food started growing, grain coming out, we had to store these grains for our future need. And we stored them. When we stored these grains, they acted as bait for the rodents, infected rodents, to reach these stored grains. And then when they reached those grains, they brought infection along with them. And again, it bridged the gap between human and animals. Deforestation, again, is the biggest culprit. And because we know that we had to resort to deforestation because we meet the growing demand of food, And it was also fueled by our need for more farmings and need for getting logs from forest. If I talk about Asia alone, it is 80.5% of the Asia is forested. And between 1990 to 2005, around 5% of the Asia forest cover has been lost. In Southeast Asia also, the forest cover has been lost. And if you to name some countries, then I would say Indonesia lost about 18% of the forest cover, Malaysia 29%, Myanmar 10%, Cambodia about 30% of the forest cover.
So looks like there's a connection trouble with Dr. Randir. Let's wait for a moment for him to come back.
Is it connected? Is it connected again?
Okay, doctor, we can hear you again. So you can continue your presentation.
Should I go back or should I continue from where I was? Which slide I was talking?
Yeah, you can continue from the presentation that stopped.
So I was talking about the climate change. Climate change again is a big culprit, and annual temperature has increased about 0.6 degrees centigrade per decade over the last 100 years. Okay, and weather events, they have increased in number, they have increased in intensity.
Doctor, excuse me, can you share your PowerPoint? It's not yet. Yeah.
Wait.
Thank you.
Now, I was talking that the climate change has again played a major role, and the temperature has risen over the last many years, and weather events have increased and intensified. The increase in river temperature or the length of the seasons has affected geographical distribution and density of species, and it has particularly influenced the transmission dynamics of vector-borne infection. Then the human-animal interface or interaction further increased when we had to switch this food demand to animal cooking. We resorted to factory farming of animals. Animal density has to increase to get more output. With minimum output, animal welfare was affected, animal immunity was affected, and then it also involved use of antibiotics to protect our large herds from getting disease and in order to achieve the desired output. Drug resistance again is an important issue that has fueled the development of disease, because the irrational use of drugs or presence of fake or counterfeit drugs has made even unopportunistic person as opportunistic, and newer diseases are emerging. And we are looking at a scenario where we may end up with a new antibiotic available to treat the infection. Population growth, crowded settlement, and urbanization is closely related to emerging and re-emerging diseases in many ways, as high human density allows diseases to spread faster. Asian sub-region has a population of about 4.7 billion, which is about 59.1% of the world population, which is 8.04 billion. Population growth in Asia has risen to 210% since 1955. In this region, Asian regions, we have highly populated countries. We have countries which are densely populated. And if we talk about the sub-regions which are present in Asia, then Southern Asia has the maximum population of 2.2 billion. It is followed by Eastern Asian countries and then Southeastern Asian countries. And then exodus of people from rural settings to urban area has also been seen. And it is seen that 51% of the people, they have, reside in urban areas in pursuit of a better life and job prospects. Then increasing tourism has also played a role in transmission of infection from one country to another. We know migratory birds. They are reservoir of several viral infections, to name few like influenza, West Nile virus. These bird philo established migratory, but just imagine if they find their home where they normally migrate to is not safe or not good to live anymore, then they can tend to change their migration. They can spread infection to your country. Over the last year, we have seen advent of new technology, especially in biotechnology, genetic engineering, and reverse genetics, which has arsenal us with techniques to make non-pathogenic organisms as pathogen. And this was even talked, rumored in case of COVID-19 outbreak also, when it was suggested that this virus has not come from wild, but it has spilled over from lab where it was scientifically treated. Now, if you look at the outline of the chronology of some of the pandemics that have been reported in the past, then in 21st century, we have seen more number of pandemics coming up. And if you compare it with the pandemics that have been reported from 13th century and what they are related. Now, this could be because, of late, if we have seen new technologies and new techniques coming up, which has provided us with benefits to detect disease easily and disease fast. If you look at this slide, we can see that there are several diseases which are lined up, or they are coming up, or they are waiting to resume technical pushing. COVID-19 has already done that. The threat of Nipah virus, isolated cases of disease are being reported. Ebola virus, Zika virus, MERS virus. And if you look at those diseases which are found out, they are very small. And even WHO, joint separately, has said that in future we may have a disease that is coming up on disease X. Famous Time magazine has also provided space to on its cover page to various diseases that have affected humans. And this magazine is a famous magazine which publishes important personality. But you can look at the cover page of this magazine, which has highlighted several diseases that have affected us. Now, if you look at this image around, you can see that since 2003, several new diseases has emerged around the world. And the new disease has emerged more in African and Asian continent. As far as this graph in this chart from Irving, again, we can see that the majority of the diseases, they have come up in Asian and in African continent. Asia, again, as suggested, discussed, is considered hotspot of emerging disease. We have high biodiversity, providing wide range of foods and vectors present there. High human density, high population of animals, and then we have wildlife trade, consumption of wildlife food, rapid urbanization and change in land use, and climate change patterns that has altered ecosystems and vector behavior promoting spread of human diseases. Now, some of the important viral zoonotic diseases reported in Asia. These are 6 viral very important zoonotic disease. MERS coronavirus, SARS coronavirus, SARS coronavirus 2, avian influenza, Zika virus, and chikungunya virus. If you look at the diseases, then you can see that their reservoir host has been violent. And from there, they have spilled over the intermediate host, and then from there, now, to talk about some of the important diseases, like the first one is severe acute respiratory syndrome. This is again a SARS coronavirus. This disease was first reported in February 2003, and a single case in which were attending a wedding function. When they left the wedding, the guests, when they departed, they were carrying this infection, and the disease spread to 5 countries within 24 hours. Within 7 to 8 months, disease spread to 30 countries, and there were 8,400 cases and now 812 deaths. Then Nipah outbreak in 1998. Just to update the audience, on Sunday we had a mortality due to Nipah outbreak in a young kid or young adult who has visited the forest area, and it was said that these people, the poor people that visited the forest, they ate wild plum food, which was contaminated by the bat. So this Nipah virus outbreak was reported in Malaysia, and it was associated with pig, and pigs got contaminated from food bags. Now, Malaysia is a Muslim major country. They don't consume pork, but then they realized the importance of exporting pork to neighboring countries, especially China, which had high demand of this crop. So they resorted to intensive pig farming, and for this intensification, they cleared forest, and when this pig farming was done, close to those farms, there were fruit orchards which attracted fruit bats, and these fruit bats then contaminated the environment, and we got infected, and from there this disease spilled over to the human. And it spread to 5 states in Asia, and it also spread to Singapore, and it resulted in 100 deaths. In order to contain the disease, around more than 1 million pigs was slaughtered. This disease has also been reported from Bangladesh, and where the palm sap was responsible, which was contaminated from fruit bats, has also been reported from fields where the coins were the intermediate host, and they transferred the infection to whom. We have had several outbreaks of influenza, and history is replete with example. We had Russian influenza, we had Spanish flu, and then among these flus, the one that comes to our mind is the avian influenza. Highly pathogenic influenza virus outbreak occurred in 1996 in China. The infection was transferred from wild waterfowl to the geese, and then outbreak occurred in Hong Kong in 1997. First human case was reported from Hong Kong in 1987, and in 2003, this strain re-emerged and caused several in Southeast Asia and Asia. India has also reported outbreaks of avian influenza. The first outbreak was reported in 2006, and then it was followed by outbreaks in 2021, where H5N8 was the strain responsible for this, and 8 states, the one shown with the red color, they were affected, and other adjoining states were put on high level. Recently, WHO confirmed a human case of bird flu in India. 4-year-old kid was infected, and it was H9N2 strain. This was in June this year. And this is the second case of infection of H9N2, and the first was reported in 2019. Then this, we have another disease of Casino forest. This is restricted to India alone. and it is present in one state, in the southern state of Karnataka. The forest, and this disease is called as Chiasenio forest disease. This disease cycles in pigs and primates, and whenever the human has encroached upon as these cycles, then disease is spread to humans, or from humans, or from these pigs to domestic animals like cattle, and from there. But then we, through strict measures, through strict surveillance and strict preventive measures, we have managed to control the spread of this disease, and it has not spread to a large extent. Then again, ribicide is an important zoonotic disease, and mostly poor rural communities in Asia and Africa is affected. Disease has not been seen in North America, Europe, and Australia, but it is prevalent in more than 150 countries. Every year, 60,000 deaths are reported, and highest death burden is there in India. One-third of the deaths there come from India, and every 9 years, someone from this countries. Then brucellosis, which is I would say is a neglected disease, and it is prevalent in not many countries. In India, also this disease is endemic, and cattle and buffaloes they have observed to be carried or affected, and prevalence level of around 16 to 18% has been reported. This is responsible for huge economic losses, and humans also, especially those in contact with these animals, or veterinarians or occupational workers, and those who deal with animals, they are at higher risk, and those people, they have also been found to be carrying the infections, and the prevalence level in those individuals ranges again from 15 to 20%. Now, what are the challenges in water prevention? The first one is the porosity of Asia is vast. Porous borders make it challenging to regulate the movement of animal products and bacteria, facilitating the spread of zoonotic pathogens across the regions. Then inadequate surveillance and monitoring is very important, because surveillance systems, they are the foundations on which disease control system sit. And these surveillance systems, they are helpful in anticipation or predicting emergence of diseases. They help us in supporting or designing outbreak response. They help us in facilitating monitoring and evaluation response. So inadequate surveillance and monitoring structure, especially in resource-limited country, is a big challenge in control and prevention of diseases. This is again compounded by cross-sector coordination, lack of cross-sector coordination. We know that we cannot see human, animal, environment, plant in isolation. They are all interconnected. They are part of the ecosystem. And when we see them together, then it is possible. But if you work in silos in different areas, like animal health expert and work in their own area, animal health expert, they work in their own area, then it becomes very difficult to challenge these diseases. Then cultural and behavioral factors, they are also important. Cultural practices, they are big hindrance, and these cultural practices varies from country to country. Dietary habits varies from country to country. They are challenging. They are big challenges compared to countries. Socioeconomic factors. If you are a poor nation, if you are resource limited nations, and you rely on animal husbandry, then your interaction with animal is low, and the chances of disease spread is low. Now let us discuss what are the key issues and way forwards. Emerging infectious diseases or zoonotic diseases, their control, it's important. It relies on integration of surveillance capacities across human, animal, plant, or environmental sectors. So integration of these all sectors is important, and use of modern technology, harnessing modern technology will help us in deeper insight into the transmission dynamic, surviving of these organisms in nature. It can help us to design preventive measures in advance. Then this One Health high-level expert panel. This is a new document, and this lays emphasis more on prevention. And in order to achieve those aspects, it relies more on or emphasize more on risk reduction activities, understanding the dynamics in natural host and environment, and addressing the human activities or anthropogenic drivers of the disease emergence. And then also uh, wants integration of several systems. But if the disease spillovers to human population, then we need to have a greatest responsibility in order to limit the spread of infection with good laboratory scope, good healthcare structure, a better investment on vaccine and therapies, and a long-term management impact to reduce the impact, long-term impact of the disease on human. International agencies, World Health Organization, FAO, World Organization of Animal Health, they have come together and they have established a tripartite guide to address zoonotic disease in countries and support collaboration between animal and humans.
Doctor, can you hear
Then,
my voice? Okay, doctor,
hello?
you have 5 minutes
Hello,
left.
Ken.
Okay, thank you.
The Food and Agriculture Organization of the United Nations, World Organization of Animal Health, and United Nations Environmental Protection Agency, they've also come together as a quadripartite organizations and have developed One Health Joint Action Plan to address these emerging and existing diseases. Then we have international health regulation framework, and this international health regulation is a legal instrument and legally binding state parties, which is aimed to help the international community prevent, protect, control, and respond to disease independent of their region, and that have the potential to cross borders and threaten people worldwide. Then we have several ongoing international surveillance efforts. One is Global Outbreak Alert and Response System. This alerts of disease or diseases or diseases. Then we have a program for monitoring and nurturing diseases, which probably largest publicly available system conducting global reporting of infectious disease and outbreak. We have World Animal Health Information System. And then there's a global variant project which is working on to look for potential viral threats to human health. And then we have the GLUE, this is a global army system for animal for health threats. And then we have a network of the OIE and FAO for influenza. Then there is one cross-border surveillance response that is existing in Southeast Asian country, which is the Mekong Basin Disease Surveillance Initiative. This was established in 1990, and Cambodia, Laos, Myanmar, Thailand, Vietnam, China, they are part of this. And this is also supported by Southeast Asian Region Office and Western Pacific Region Office, and facilitate cross-border cooperation and surveillance for control of infectious disease. India is also very active in One Health initiative, and we have several committees and organizations that are looking after these sectoral collaborations to manage any emerging or emerging diseases. India has also launched Animal Pandemic Preparedness Initiative and a health system support for health to look after or address any animal pandemic and preparedness to handle any unknown diseases in the future. We have a national animal disease referral expert system, which collects disease data from different states. It also collects other demographic information and better information, meteorological data, analyze this data, and then issue one disease one alert message for different states to take preventive measures. Now you can see our prime ministers who country recently they had the privilege to use the G20 summit, and India was the president of it, and The motto of that summit was also One Health, One Problem. India has established National Institute of One Health in Nagpur, and this is a dedicated center for research in One Health and enhances the national capacity to deal with epidemic of all potential respiratory diseases that may affect regional. To sum up my presentation, I would say that diseases do not respect national borders and can rapidly spread across regional periodic evaluation of surveillance capacity and identification of capacity strengthening, especially for the emerging disease and this is the challenge, and we have to meet that challenge. There is a need of collaboration between public health, community, veterinary epidemiologists, data specialists, physicists, disease modelers, scientists, economists, environmentalists, to work together so that we can support each other efforts to contain the disease. The level of One Health education program should be increased. It may also involve educating or aware the population about various diseases, their cause, their transmission, and so that we can promote healthy behavior and practices that can prevent that spread and of virus in the disease. Then, to reduce the spread of virus, this is in Southeast Asian region, is a multidisciplinary network of countries are important. We know that some social factors or cultural factors of the regional disparity may be a challenge, but then it is important to work on that. So that therefore can implement those measures and prevent the disease outbreaks, and we can design measures which are tailored to the region-specific needs and circumstances. And at the end, it is also important that we have our political establishment handholders provide support, provide finances, so that we can take up this challenge and we can create a better world, or we can create a better environment for our future generation. Thank you all for your time, and I acknowledge the Pankaj Sakka for compilation of this presentation. Thank you very much.
Okay, thank you so much, Dr. Andir, for a very nice presentation. So, Doctor, in here we already have some question for you. Okay, can you hear me clearly, Doctor?
Yes.
Okay.
Yes, I can.
So the first question is, how can regional cooperation and international partnership be strengthened to enhance border control measure for zoonotic disease prevention in Asia? This is question from our student.
Yeah, it is important that these all I have given an example of background placing data disease surveillance program. Like this, we can these countries can identify important diseases and design measures. And while designing these measures, it's important that any cultural, any social differences that may hinder those challenges, and that at the same time, international agencies such as WHO and World Organization of Human Health, International Animal Protection Agencies, and occasional offices of WHO, they are always ready to handle and supporting our efforts to control disease.
Okay, so we have more question for you, Doctor. So the next question is, what innovative technologies and approach can be employed to improve surveillance and then early detection and response to zoonotic disease outbreaks at national borders in Asia?
I have gone through some literature here, especially if I take the example of the data surveillance system. Here, the countries at the border, they have established screening points where both countries operate. And over the period of time, we have seen that new diagnostic techniques that they have been constructed. If I take the example of COVID-19 outbreak, within a period of 19 months, we have a huge data of different strains that have come up, and that was possible through whole genome sequences. And we have several tools available where the huge data that is being generated can be analyzed, and these analyzed data can be used to generate predictive response. We can predict the course of disease, and we can take screening measures to prevent the spread of the disease.
Okay, so I think we have the last question. It's also from our student. Uh, so the last one is, what are the primary challenge faced by ASEAN countries in preventing and controlling the spread of zoonotic disease across the borders?
Again, it's a big continent, right? So we are home to almost 60% of the total population. We have several countries, and these countries are diverse. They have different cultures, they have different social factors, they have different dietary factors. So we have to look after those things, and we have to respect those differences. And at the same time, we have to see that whatever measures or service method we adopt, that respect all those issues. And we take into consideration unified or accepted protection measures which are internationally accepted. Can help us to make better decisions, arrive at a decision where we can decide which way to move in order to reduce the impact of
Okay. Okay, thank you so much, Dr. Randir Singh, for your time. And before we close the session with you, we have some token of appreciation for you. So this is the certificate from the Universitas Airlangga, and the certificate is for you. Thank you so much, Dr. Randir. We hope we can see you again in the next event. Thank you, Dr. Randir. Okay, so we will go to the last presenter. We will continue with Dr. Ugbo. Dr. Ugbo, are you still here? Or can you hear my voice? Okay, so Dr. Ugbo already come back. So, Dr. Ugbo, we will share your PowerPoint. So we will continue with Dr. Ugbo, the last PowerPoint that we shared. Okay. Okay, Dr. Ugbo, the time is yours.
Yeah. Hello, Doc.
Yes, we can hear you
Hello.
clearly. Yes, hello, doctor. You can continue, doctor.
Can you hear me?
Yes, yes, we can hear you.
Can you hear me?
Yes, doctor, we can hear you.
Okay, thank you. We have to restart back, please. You have to start the. Can I share the PowerPoint myself?
From what part, doctor?
Can I share the PowerPoint?
Okay, okay.
Hello, Doc.
You can share it.
Can I share the PowerPoint by myself?
Oh, you want to share it by yourself?
I can share it.
Oh, okay, okay. So Dr. Bo will share the PowerPoint by himself.
Yeah, because I'm not seeing clearly what you are sharing. It's not too clear.
Okay, doctor.
It's not too clear. So let me share it by myself. Thank you.
You can share your PowerPoint, okay?
Your own is still showing. Okay.
Okay, doctor, you can share your PowerPoint. Okay.
It's okay.
Okay.
So first of all, let me apologize for the network glitch. I'm sorry for that. I pray it will not happen again, please. So I am making a presentation, challenges, future direction. Let's look at what are the antimicrobials. Antimicrobials include antibiotics, antiviral, antifungi, antiparasites that are medicine used to prevent and treat infection in human and animal and plants. Therefore, the effectiveness today is now in jeopardy because of number of the antimicrobial agents that once worked no longer do so because microorganism has become resistant to them. And antimicrobial threaten countries in different ways and in varied directions. Now, antimicrobial resistance. What do you understand by the word antimicrobial resistance? Antimicrobial resistance occurs when bacteria, virus, fungi, parasites change over time and they no longer respond to the medicine that we are being used to treat before. So antimicrobial microbial resistance makes infection hard to treat and increases risk of disease spread and severe illness and even death may even occur as a result of antimicrobial resistance. And today, who has declared a threat facing humanity? Now, looking at different types of antimicrobial resistance, we have what is called multi-drug resistance. Multi-drug resistance is known to occur when non-sensitive to at least one agent in 3 or more antimicrobial categories occur. And we have the second one called extreme drug resistance. This one occurs when there is no sensitivity to at least one agent in all categories except in 2 or less. And the third one is pan-drug resistance, which occurs when there is non-sensitivity to any agent in all the antimicrobial categories. There are a lot of factors that can contribute to antimicrobial resistance. One of the factors are environmental factor, drug-related factor, patient-related factor, and physician-related factor. Looking at environmental-related factor, one of the major contributors of environmental-related factor is overcrowdedness, poor sanitation, ineffective infection control program, or increase in national and international traveling. Take for instance, if someone that is a carrier is traveling from one region to the other region, he can actually disseminate antimicrobial resistant organisms to the next country that is crossing over. Another one is widespread of antimicrobial in animal use, in husbandry, agriculture as a growth promoter. Today, you can believe in me that a lot of people use antimicrobial as a great promoter to enhance the growth of their animals. The next one is drug-related factor. Fake drug contributes to antimicrobial resistance. Quality of the drug. If there is a low concentration of a particular drug, it can actually cause a resistance because the actual concentration that is supposed to inhibit the growth of the microorganism is no longer there. So it cannot actually work or actually inhibit the growth of that particular organism that is causing that infection. Over the availability of antimicrobial. A lot of over-the-counter antimicrobial are being disseminated all over the world. People go to over-the-counter, collect drug unprescribed, and start using. They also encourage antimicrobial resistance. and irrational fixed dosage combination. Sometimes we combine dosage of drug without actually looking at if it is actually going to increase synergic effect or actually decrease the effectiveness of the antimicrobial agent. Next one is the patient-related factor. Poverty is one of the factors that are actually creating antimicrobial resistance. Poor concept or lack of sanitation, self-medication. Some people intend to medicate themselves when they are sick. They go to over the counter, collect antimicrobial without following a doctor's prescription. Poor enhancement of dosage regimen. Some people don't actually follow the dosage regimen prescribed by doctor. They just, maybe once you get, you feel you are a little bit okay, you now drop the drug and feel you are okay. All those things encourage antimicrobial resistance and lack of education. So when people don't have knowledge about antimicrobial resistance, they keep on abusing drugs, thereby increasing the rate of antimicrobial resistance in the environment. The next one is prescribers and physicians related factor. Inadequate dosage also is one of the major factors, and inappropriate use of available drug. Sometimes doctors intend to prescribe blindly without sending a patient to proper text. They will just prescribe drug and give to the patient. All these things encourage antimicrobial resistance. And next one is overusage of antimicrobial. Once a particular antimicrobial has been abused and overused, it also creates resistance. Microorganisms became used to it, and they form resistance against that particular organism. And lack of training and current knowledge. Some doctors don't intend to follow up with the current trend of knowledge on antimicrobial resistance and also treatment of infection. Now, there are some modes of spread of antimicrobial resistance. Take, for instance, sometimes when our animals are sick, we intend to treat them with antimicrobial agents. Sometimes we don't actually allow the animal to get healed completely and the residue of those antimicrobial agents gets rid from their body before selling the animal to market. Sometimes some of the residues are still inside the animal. They will now sell to the market. People intend to slaughter and consume, thereby consuming the residue, and it also encourages antimicrobial resistance. There are so many mechanisms of bacteria can use to project resistance. So one of these mechanisms is one, limit uptake of drugs and modification of target site. If you look at the structure here, you can see where the organism is modifying its cell wall to enable the antimicrobial agent not to penetrate. And another one is inactivation of a drug. They inactivate it by hydrolyzing this antimicrobial agent. They will no longer be effective. An active drug pump. Under active drug pump, these organisms intend to push off the drugs that comes into their cells, thereby reducing the concentration that's supposed to inhibit the growth of this particular antimicrobial. So globally, it has been estimated that over 1.2 antimicrobial related infection checked, if it is unchecked, by 2000 deaths will be attributed to antimicrobial related infection. So current global impact of antimicrobial resistance, it has been estimated that in every one child that died, in every 3 minutes, it is attributed to be as a result of antimicrobial related. And they stated, if it is not properly checked, you see, time will come, over 28 million people will be pushed to abject poverty as a result of antimicrobial resistance. There are some other driving factors that can contribute to antimicrobial resistance based on One Health. One of the driving factors is misuse of antimicrobial in human. And the next one is misuse of antimicrobial in terrestrial and aquaculture animals. And also poor infection and disease prevention control in food and feed of animals. And misuse of antimicrobial in plant and crop production. And sometimes the discharge of the waste from healthcare facilities and pharmaceutical manufacturer environment are being pushed into the environment. All these things accelerate antimicrobial resistance. Now, how do we manage or respond to antimicrobial resistance? Environment plays a key role in developing a transmission and spread of antimicrobial resistance. Therefore, the response must be based on one health approach, recognizing the human, animals, plants, environment as interconnected and indivisible at the global level, regional and local level for all sectors and stakeholders in order to prevent all this antimicrobial resistance. So prevention is still at the core of the action needed to hit the emergence of antimicrobial resistance. And the environment is the key part of the solution. So we cannot actually eradicate antimicrobial resistance without studying and looking into our environment inward. So there are a lot of approaches on antimicrobial approaches for combating antimicrobial resistance. Now, we need to increase collaboration on the area of antimicrobial resistance. And also regional drug usage. You need to be noted the kind of drug that are being used in a particular region and also the rest of this. And also rational usage. You don't just come and use drug anyhow without following the controls. It's very, very important. Microbial surveillance, very, very important. Educating and motivating people. And there should also be a kind of research and development of drugs and even vaccine. Immunization also is very, very important to cover a wide range of places that is being suspected to spread antimicrobial resistance. Whenever we go out and come in, it is very, very important that we observe hand washing to reduce the microbial contaminant in our hands to avoid us importing a pathogen from one area to our environment. So new programs need to be done on antimicrobial resistance. An antimicrobial resistance committee need to be created, functional ones, not just a committee. And there should be a serious national policy to follow in order to combat this antimicrobial resistance. So there are some strategic strategies to follow for combating antimicrobial resistance. Number one, strategic area one, awareness and education. Improve awareness and understanding of antimicrobial through training and education. Microbiologists, epidemiologists, and public health officers should know the basis of antimicrobial activities, basis of antimicrobial resistance, source of antimicrobial mechanisms, transmission route, and magnitude of the program in the society. Best way of using a laboratory test for patient care, not just you come and use any type of a laboratory test. There should be a best way of detecting a particular resistant organism. Then the strategic area 2 is surveillance and research, very, very important. Strengthen knowledge and evidence through surveillance and research. Microbiologists and epidemiologists and public health officers are responsible for quality diagnosis and surveillance, production of data, collection, analysis, and report source of the resistance and source of resistant mechanism, very, very important. They also have to go into detection, confirmation, characterization, and communication of antimicrobial emergency, microbial resistant emergency. They also measure the impact of antimicrobial resistance in the society through research. So the strategic area 3, that is infection, prevention, and control. Reduce incidence of infection through effective hygiene, infection, prevention, control measure through 1. Under this infection, prevention, and control, effective hand washing is very, very important. Cleaning and sterilization procedure also need to be considered, and reduce healthcare associated infection. So all these things need to be come together in order to combat this antimicrobial resistance. Prevention at community level, vaccine, hand washing, and environmental sanitation, very, very important. Under animal health prevention and control, vaccine and biosafety and hygiene are very, very important, and also sustainable animal production. Strategic area 4, appropriate usage of antimicrobial agent, optimize use of antimicrobial in human and animal. So it is always very, very important that the microbiologists know the timely, appropriate, and quality diagnostic results. And they are the ones that have to maintain all those in the data and select the report on sensitivity according to each pathogen and site of infection and antimicrobial profile, and report based on pharmacodynamic and pharmacokinetic parameters. So special tests when necessary, such as rapid testing, quantitative testing, bactericidal testing, evaluation of synergic activity, and rest of them. And antimicrobial surveillance, very, very important. What are the challenges? The possible challenges and challenges of antimicrobial resistance. What are the challenges? Massive and irrational usage of antimicrobial in human and animal and plant is the very much challenge that is facing antimicrobial resistance. And poor and under-regulation of antimicrobial usage in low and middle income countries. In these low and middle income countries, sometimes the antimicrobial usage are not actually being regulated. People use antimicrobial how they want. They go over to counter to buy, whether prescribed or non-prescribed. They buy whatever thing they feel they can use to treat themselves. It's one of the challenges facing this antimicrobial, combating this antimicrobial resistance. And antimicrobial accessibility without prescription by qualified health professionals. Rampant contamination of standardized and fortified medicine into the supply chain of antimicrobial agents. These are one of the problems. People intend to import fake drugs, mix it together with standardized ones. So, and all of them look alike. You may not actually detect which one is fake and which one is not fake. It's also one of the greatest challenges. A massive usage of antimicrobial in the animal husbandry sector, including aquaculture for growth promoter and treatment or prophylaxis. So what are the future directions for combating this antimicrobial resistance? One, development of better rapid diagnosis tool treatment option. Second one, increased collaboration between government, non-governmental organizations, professional groups, and international agents in area of antimicrobial resistance. And establishment of new network that undertake surveillance of antimicrobial usage and antimicrobial resistance. Establishment of international approach for control of counterfeit antimicrobial agent is very, very important to combat it. Those are the future directions. And promoting research and sustainable investments and inactivate for incentive for the research and development of new drug and vaccine. It is very, very important that there is always an incentive to encourage researchers on the area of developing new drugs and vaccine, forming new and reinforcing existing program to contain antimicrobial resistance. There should be a new program formed and also reinforcing the existing program to enable this antimicrobial resistance to be the program to get across to the society for people to know the impact of this on the society. So in conclusion, antimicrobial resistance is a complex problem with many diverse contributing factors. Hence, prevention is still the best tool to combat antimicrobial resistance. Now, rational usage of existing antimicrobial drugs, development of new effective compound, and new diagnostic technology is the major need right now. Functional and comprehensive global national antimicrobial policy, resistant policy, should be developed and implemented by involving policymakers, partners, stakeholders in public health. Establishment of local, national, and regional policy platform for management of antimicrobial resistance could play a crucial role in combating antimicrobial resistance. Continuous collaboration of World Health with Food and Agriculture Organization of United Nations and World Organization for Animal Health in One Health approach is to promote the best practice to reduce the level of antimicrobial and slow its development is strongly recommended. So these are the possible references where you can get some of this information that is in this slides for your personal use. Thank you for listening to my presentation, and thank you again for giving me the opportunity to be part of this this year Icvmhs conference. Thank you so much for listening to my presentation.
Okay, thank you so much, Dr. Ugbo, for your very good presentation. And so, Dr. Ugbo, in here we already have a few questions for you from our student. So, the first question is from Bella. So, Dr. Ugbo, what are the key strategies currently being employed to combat antimicrobial resistance?
Okay, thank you very much for that question. Like I said in my presentation, that one of the major strategies in combating antimicrobial resistance is through research, one. Second, through education. We need to educate people on the impact of antimicrobial resistance in the society. And through prevention, because when you educate people, you have to tell them, how will you prevent things you need to do? When you go out, you need to wash your hands. When a doctor prescribes a drug for you, you need to complete your regimen. So it is very, very important that we educate the society. We also make sure that the people have a wide knowledge on the use of antimicrobial resistance. People should also stop abuse, and they stop improper usage of antimicrobial in improper areas. Take for instance, the use of antimicrobial that is meant for human treatment in the treatment of animal is one of the major problems. If people are aware about all these things, not to use antimicrobial that is meant for human treatment in animal treatment, they will also know that if they stop all those things, they can actually reduce the impact of antimicrobial resistance in the society. And also, it is very, very important that there is a national policy against this, and they create a new agencies that can work in hand to disseminate this information to the society. By so doing, this can be combated from the society. Thank you.
Okay, so doctor, we have next question. It's from Zoom. So it's from Miss Ruth. Okay, can I see the question? Okay, so what is the economic incentive or policies could help encourage the development of new antibiotics or alternative therapies?
Please come again. I didn't get your question properly.
Okay, so it's from Ruth Nwodom. So doctor, what is the economic incentives or policies could help encourage the development of new antibiotics or alternative therapies?
Okay, just like I said, that one of the future way forward in combating antimicrobial resistance in the society is through research and development of new antimicrobial resistance, new antimicrobial agent or vaccine. One of the incentives is this. Government can actually build laboratories. They can actually also a kind of equip laboratory, bring in new technological new machines that has a current technology to equip some of these research centers where they are working on development of a new drug. And when there is a kind of a work towards a new drug, you have to encourage the people by giving them some little incentives, like a kind of making their life very, very comfortable. Because it's when you have a comfort life, it's where you can work effectively mentally. So if you have a distressed mental health, you cannot actually work. So they need to create a comfort life for them. Maybe when there is a kind of a new approach to development of drug, they make the people to be comfortable to complete the research, not a kind of when government will give a research, or maybe you are having a research towards development of a new antimicrobial agent and you don't even have enough funds to complete the research. In that kind of situation, government should be able to raise funds and sponsor the research for the person to complete it, because it's going to have a great impact on the society. Thank you.
Okay, thank you so much, Doctor. So maybe this is the last question from our student also. So it's from Roro. So Doctor, what is the future directions and innovations are being proposed to address antimicrobial resistance effectively?
Okay, just like I said, one of the future direction is research towards a new development of new antimicrobial agents. Do you understand? That is one of the major future direction. We have to research towards development of a new antimicrobial agent that can be a kind of a broader antimicrobial and agent that can actually inhibit both gram-positive and even can actually inhibit, have some activities to other related organisms, not a kind of having a particular antimicrobial agent to hit the resistance of a particular organism. They can develop an antimicrobial agent that can have a wide range in inhibition of different kind of organisms. Is one of the way forward. Then another way forward is making sure that the people in the society actually follow a proper hygiene in order to prevent this particular, because after in all these things, prevention is still the major way forward. Because if the people abuse drug, if they are not educated to know that abusing of drug is one of the major ways that can encourage resistance. Because if you look at some of the nations like Australia, where drugs are not highly abused, you can bear me witness that antimicrobial resistance is not too strong in those areas. But in Africa and some part of Asia, where people go to over the counter to buy, you see there are a lot of antimicrobial resistance issues in those areas. So I think prevention is still one of the major way forward, and development of a new antimicrobial against those ones that are already resistant. Thank you.
Okay, thank you so much, Doctor. I very agree with you for that point. So, yeah, prevention is better than treatment. So, Doctor, before we close the session with you, we have some token of appreciation for you. This is the certificate from our university, from Universitas Airlangga. Thank you so much, Doctor. I hope I can see you again in the next event. Thank you so much for
thank you. I appreciate. Thank you.
So
thank you for the opportunity given to me to meet you again through Zoom. Thank you. I appreciate the care people took to me the time I came to Elanga. I really appreciate all of them. Thank you for having me again.
Okay.
I'm glad to meet you guys again.
Okay. Thank you,
Appreciate it.
Doctor.
Thank you.
Okay, so this is the end of the session in the room number 1. We still have ongoing session in the room number 2 and also room number 3. And not only that, we still also have some session for the oral presentation in the room 1, 2, and also 3 for the oral presentation. So if you would like to join another session, you can just join another link that we already share in the website or in the group chat. So this is the end of the session for the room number 1. And thank you so much for your attention and have a good day. Thank you.
Once I is the network.
Welcome back, everyone. That sums up our main event for today. Because everyone has done such splendid presentations and posters, as a token of our appreciation, we would like to award representatives for best posters and oral presenters that will be announced by Professor Dr. Erma Safitri, Doctor of Veterinary Medicine, Master of Science. We welcome Professor Erma. The time is yours.
Oh, nggak kelihatan.
Welcome back everyone. So sorry for the previous technical difficulties because everyone has done such a splendid presentation and poster. As a token of our appreciation, we would like to award representatives for best posters and oral presenters. That will be announced by Professor Dr. Erma Safitri, Doctor of Veterinary Medicine, Master of Science. We welcome Professor Erma. The time is yours.
Ladies and gentlemen, it is my pleasure to announce the winners of the Best Oral Poster Presentation at the 4th International Conference on Veterinary Medicine and Health Science 2024. The selection committee had a very challenging task, facing a multitude of insightful presentation throughout the conference. We were particularly impressed by the depth of research, clarity of delivery, and engaging visuals in the winning presentation. Our congratulations goes to the best oral presenters. First, Muhammad Rafiansa Jawindra SKH. Second, Didi Tulus Subekti Dr. Hewan MSc. And then Mr. Fatah Nugroho Dr. Hewan. Congratulations. Okay. We were also trying to congratulate the best poster presenters. Saifur Rahman, Anggun Hoirun Nikmah, and then Satria Tegar Rahmadani. Congratulations to all of the winners. The organizing committee will be contacting the winning presenters shortly to discuss prize details and next steps. We extend our sincere gratitude to all presenters who shared their valuable research and fostered stimulating discussion throughout the conference. Your contribution were instrumental in making this event a resounding success. We look forward to seeing you again at the 5th International Conference on Veterinary Medicine and Health Science. Thank you.
Thank you, Professor Erma, and congratulations for all the winners.
Now, we've reached the end of today's agendas. Thank you to all the honorable guests, speakers, and participants for attending today's event.
With that, our duty as the Master of Ceremony for the 4th International Conference of Veterinary Medicine and Health Sciences from the Faculty of Veterinary Medicine, Universitas Airlangga, has come to an end. Once again, my name is Naura Kalila Sandrina.
And I'm Kiara Tanakirana. See you next year
at the
5th International Conference on Veterinary Medicine and Health Sciences.
wisata liburan pertama, kado dari mama dan papa, karena berhasil naik kelas, walau rata-rata pas-pasan. Nekat pulang lewat jam malam, dilarang jalan-jalan sebulan. Setahun bimbingan belajar, gawat ujian sekolah. Serpihan berbahaya